We couldn't see the live.
Recording in
Thank you. Thank you very
progress.
much. Thank you very much. So, good evening, all. Good evening, everyone. Welcome to this exciting evening as we kick off the iHera webinar. And it's true pleasure to see many esteemed embryologists, IVF experts from across the world gathered here on this platform. So, you are the composer and inventor in the field of reproductive health, making invaluable contribution to innovative medical treatments that are transforming lives nationwide. So we are at UNS. We take a pride to being one of the fastest growing nutraceutical company, and we are committed to making a significant impact on people's lives through our high quality products. We are having our male fertility and female infertility portfolio, which includes CoQ 300, CoQ LC, CoQ 4, CoQ FEM, and CoQ MAN, which is the design to empower individuals on their journey to become a parenthood. Now, it's my honor to invite today's coordinator, Dr. Nancy Sharma, to introduce today's distinguished speakers as well as to set the context for our discussion. Yeah, over to you, Dr. Nancy.
Thank you, Nakul. And good evening, everyone, and a very warm welcome on behalf of our team iHera. And today in our session, we are going to discuss about the cytoskeleton system of oocyte. We will see how cytoskeleton plays a role in impacting ART outcomes. And for that, we have wonderful speakers and a beautiful moderation team with us. So before taking much of your time, I'll start with today I'm delighted to be moderating today's session alongside Dr. Nidhi Yoshita Tanwar and Dr. Ananta Lakshmi. Before we begin, I would like to call our esteemed founders and co-founders on board, Dr. Ved Prakash sir, Dr. Sanjay Shukla sir, and Dr. Charudhar Joshi sir. Sir, Dr. Charudha Joshi, sir, I want you to please come on stage and say a few lines for today's webinar and enlighten us with your thoughts.
Yeah, thank you, Nancy. It's again a pleasure to invite all of you on this platform of iHera, which is a non-profit platform for offering services and opportunities to the young upcoming people to share their knowledge, scientific events. And now, as we know, today we are focusing on the gametes. And as an embryologist, it is always a challenge to see the quality of oocyte, quality of sperm, how they are going to fertilize. And then, irrespective of many times we see that irrespective of everything is perfect, all right, still there is less fertilization or no fertilization. So today this webinar is focused on the interiors of the gametes, particularly the cytoskeleton of oocytes, because we all know the life starts with the oocyte, fertilization happens, then embryo formation starts. So we have distinguished speakers with us, they will be discussing with us about the structure, about the cytoskeleton, about the abnormalities, and how we can judge, or we can manage, or we can just assume that this may be a problem with this oocyte and we can try to rectify it. So I think this is going to be a wonderful event, wonderful webinar, and all of us will have good insights of the gametes to learn about. So let's hope for the best. Thank you, Nancy, and you can proceed with the further proceedings.
Thank you so much, sir. So without further ado, I think we should start with our very first lecture, which is going to be on the basics of cytoskeleton system in oocyte, for which we have an excellent speaker with us, Dr. Sindhuja Srinivasan. She is IVF lab manager, academic coordinator at Sri Chakra Fertility. She's also SRM certified. She's working with many communities and executive committee member of TAP ISAR 2024 to 2026. Also a part of Youth Brigade ISAR 24 to 26. She is having many achievements under her name. I would like to call Dr. Sindhuja on board. Welcome, ma'am.
Thank you, Nancy, for the kind introduction. Can I share my screen? At the outset, I'd like to thank IHIRA for inviting me for this talk. It is excellent to see such great work being done. And thank you to Charudat sir, Ved sir, and Sanjay sir for moderating this session. So with the chair's permission, can I start my presentation?
Yes, please go ahead.
Thank you, sir. Okay. So it is a very interesting topic that's been given to me, which is the basics of cytoskeleton system in the oocyte. So first, let's see what is a cytoskeleton. So it is a structure that helps cells maintain their shape and internal organization. It also provides mechanical support that enables the cell to carry out essential functions like division and movement, and several different components. So when we say cytoskeleton, it's not just one part of the cell. It is several different components which work together to form the cytoskeleton. So we're going to be starting with very, very basics, and then we're going to see how it correlates with the oocyte. So what are the functions of the cytoskeleton? Why should we know about these structures? So basically, it gives shape to the cell. They're responsible for contraction. They help in the movement of the cell and movement of the organelles within the cell. It helps in cell division. It helps in the intracellular organization of the cytoplasm. It helps in establishment of cell polarity and other functions which are necessary for maintaining the homeostasis of the cell. So key functions are to maintain structure, transport as in inside and outside the cell, and also organelles within the cell, and for cell division. So the cytoskeleton, as I said, it's a very dynamic structure. So it's not like it's just present at one place. It is a very dynamic structure. And there are three major filaments and tubules, which are under the influence of proteins, which regulate their length, state of polymerization, and level of cross-linking. So the membrane cytoskeleton was first described in erythrocytes. We've read about this in basic biology, where it maintains the biconcave shape of the oocyte, of the erythrocyte. And these membrane skeletons, they also play diverse roles like DNA repair mechanisms, calcium release from internal stores, which is also reflected in the oocyte for oocyte activation, and for synaptic transmission in the neurons. So now what are the components of the cytoskeleton? So there are three major components which are being built over and over and over. So the three main components are actin filaments. As you can see in the picture, the actin filaments are very thin filaments. They involved in the cell shape, movement, and signaling. They're about 6 nanometers in width. The intermediate filaments, as you can see in the last part of the image, is about 8 to 10 nanometer in width, and they mainly provide structural support and stability. And we have the microtubules, which are long tubulin-based filaments. It's about 25 nanometer in diameter, and it is responsible for cell shape, motility, and intracellular transport. So all of them have very overlapping functions. We'll see in detail about them. So the actin, like I said, is very dynamic and directional. It helps in structure and movement, and it's also the tension element. An intermediate filament is relatively static. It does not move much, but it provides structural integrity. And the microtubule is very dynamic. It keeps moving. It's directional, and it is non-compressible. So this picture I've added because I want you to know that, the microtubules are generally present around the nucleus. The intermediate filaments are scattered in the cytoplasm to provide structure, and the actin filaments in a mature cell are generally in the periphery. So, and coming to the other organelles, you can see that there are centrosomes, there are intermediate filaments, there are microfilaments, there is a plasma membrane, there are lysosomes, which mainly digest food, there is Golgi apparatus, which modifies the protein, the cytoplasm, the mitochondria, which we know is the powerhouse of the cell, and there is endoplasmic reticulum, rough and smooth. Rough is associated with ribosomes and makes secretory and membrane proteins. The peroxisome, which metabolizes waste. The nucleolus, which is a condensed region where the ribosomes are formed. And we have the chromatin, and we have the nuclear envelope. So these are the major components of the cell, which I think we've read from 6th standard, but how these cells actually correlate with us in the oocyte. This is what we're going to see today. So now all these actin, intermediate filaments, and the microtubules which we saw, and all the cellular organelles, what component or role do they have in oocyte and its development? So first, let's see about the actin filaments in the oocyte. So like I said, the actin is the one that maintains the shape, and it also helps during maturation. The cortical actin network, it helps in signaling and interactions with the surrounding cells. And it also helps, contributes to the polarity and rotational movement. So let's see a bit in detail about that. So first is the polarity. So what is a polarity in the oocyte? So the polarity is basically the one which decides which is the animal pole and which is the vegetal pole, and the cleavage plane and furrow is decided based on this. And actin plays a major role in deciding the polarity of the oocyte. So how does actin play a role in that? Because certain organelles have to move to the animal pole if it has to be decided that this is the animal pole. Next, the cortical actin. So you can see that these are the cortical granules in the oocyte. So the actin is generally in a mature oocyte arranged in the periphery of the, just under the plasma membrane, which helps in the cortical reaction, which is required at the time of maturation and oocyte fertilization. And it also helps to form the gap junction and the transport of nutrients at the ulama. So what is cortical rotation? Like I said, once the sperm entry is done, there is about a 30-degree rotation which happens, which decides the plane of cleavage for the first, second, and subsequent cleavages. And for that also, actin filaments are very important. And next is, of course, in fertilization. They are involved in the cortical reaction during fertilization. I'll just talk a bit about it in detail later. So to see a little bit about the structure of the actin filament. So when the actin filaments are present free, they are monomers. It's called G-actin. And when they combine together, they form dimers and trimers. And then they form this structurally, functionally active actin, which is also called as filamentous actin or F-actin. And in order for it to happen, it has to be bound to ATP. And the actin filament also has a structural polarity. That is, it has a minus end and it has a plus end. So you can see that the plus end, when there is an action of ATP, it grows towards this end, and here it disintegrates towards this end. Now moving on to the intermediate filaments in the oocyte. So they, as I said, intermediate filaments, they are between actin and the microtubules. They just mainly provide mechanical support, and they help in organizing the cytoplasm during maturation and fertilization. And they are more stable than microtubules or actin filaments. So the major function in oocyte, like I said, cell support, mechanical stability to the oocyte, and support the overall structure and integrity during meiotic maturation and fertilization. But in oocytes, they actually have a limited role because they are not as prominent as their function in the other cells. But especially during oogenesis, when the cell size keeps increasing and more stability is required, that's where intermediate filaments actually have a major role. So to see the structure, as you can see in this image, they are made up of several strands of fibrous proteins that are wound together. And the elements of the cytoskeleton get their name from the fact that the diameter is between the two other filaments that I mentioned. So their function is purely structural. And I actually added this picture for the last line because these are the structures which bear tension. So when we kind of mishandle or trouble the oocytes during ICSI by creating this sort of a tension which is not required, it is still the intermediate filaments which kind of keep the structural integrity together. So now moving on to one of the major structures, which is the microtubule of the human oocytes, which are absolutely essential for maturation and fertilization. So they are the largest component of the cytoskeleton, and they are found throughout the cytoplasm. They are made up of alpha tubulin and beta tubulin, and both of them bind to GTP. So whenever they are bound to GTP, they then form structures like this, which forms a diameter of about 25 nanometers. And as more and more GTP tubulin dimers assemble onto the filament, GTP is slowly hydrolyzed by beta tubulin to form GDP, and then there it disintegrates. So how does this happen? Because of polymerization and depolymerization. So at this juncture, I'd like to add a very interesting point, especially if there are students who are listening. So every time when a senior tells you, don't expose the oocyte to light, don't make sure that the temperature is 37 because we should not drop by several degrees or increase by several degrees. And then many students will just say, okay, and follow it blindly. But there are some students who will ask, why? And then, we just say because it is toxic. So how is it actually toxic? So when we see in the meiotic spindle, this polymerization and depolymerization of the meiotic spindle and the action of GTP and GDP, This particular step is the one that is actually very temperature sensitive. So when we say meiotic spindle damage will happen when there is a difference in the temperature, so this is the exact step that is being affected. So in the human oocytes, like actin, intermediate filaments do not have any polarity, but actin has polarity. Actin and microtubules have polarity where they have a minus end and a plus end. So unlike actin, microtubules can actually grow in both directions, but it is more in the plus end. So what do they actually do in oocytes? They help resist compression and they provide. So when we say these organelles are being rearranged in the cytoplasm during maturation and fertilization, so how do these organelles actually move? Because these microtubules create a path for them to move. So the pull is replicated, pull the replicated chromosomes to the opposite ends of a dividing cell, and they are the structural elements of flagella, cilia, etc. And the centrioles. The latter are the two perpendicular bodies of the centrosome. The centrosome is the microtubule organizing center. So, like I said, the minus ends of the microtubules, they are anchored in structures called MTOCs, which is microtubule organizing centers. They're also called as the centrosome and is usually located just adjacent to the nucleus. And the microtubules tend to grow out from the centrosome. So, and also, as we know, the other roles of microtubules in the oocyte is spindle formation, which is the most important. So microtubules form the mitotic spindle during meiosis, and they also help in cell polarity, just like actin. And so this cell polarity that the microtubules produces is actually different from the role which actin had. So actin is helping us to decide the cell polarity of the oocyte, wherein it helps to decide which is the animal pole and which is the vegetal pole by rearranging the organelles, and also for the further cleavage patterns. But the cell polarity produced by the microtubules actually helps in the asymmetric division. For example, we know that oogenesis is actually asymmetric division, where the first polar body is not the same size as the oocyte. So in order for this asymmetric division of the cytoplasm, we need microtubules. And also for intracellular transport, which is involved in the transport of mRNA and organelles within the oocyte. And for the oocyte maturation, it is crucial for chromosomal segregation during meiosis. So at this point, we should also know a fact that all of us know, that there is during oogenesis or oocyte maturation, even though microtubules and the microtubule organizing center and all those things are present, the centrioles are not there in the oocyte. From the sperm, which comes inside during fertilization, which helps with further migration of the pronucleus and so on. So now let's briefly see, I've already told what actin, intermediate filaments, all these things do in oocyte maturation and fertilization, but just let's see oocyte maturation and fertilization separately a little bit in detail. So during oocyte maturation, what happens? So as the oocyte mature, the cytoskeleton undergoes substantial reorganization. The microtubules and the meiotic spindle. In prophase 1, the microtubules are disorganized, but as the oocyte progresses to metaphase 2, the microtubules form a spindle structure, and it is necessary for the segregation of the chromosome and alignment. And the actin filament reorganization, it is essential for the reorganization of the cytoplasm, because during the transition from the prophase 1, that is the GB oocyte, to metaphase 2 oocyte, they undergo significant changes. So you can see in the oocyte here itself, like, in the GB oocyte, how the organelles are arranged. So the mitochondria is generally around the nucleus, and the endoplasmic reticulum structures are here, and the Golgi is also very dense. Whereas in an M1 oocyte, you can see that they are starting to get scattered around, and the cortical granules also from the center, they start moving to the periphery. These black dots are the cortical granules. And in a mature oocyte, you can see how the cortical granules are arranged linearly almost in the periphery. And the Golgi break down into smaller pieces, and the mitochondria are all fully scattered in the cytoplasm. So like I said, so this is another picture which is depicting the cytoskeletal structures during the cytoplasm. So along with the organelles, the cytoskeletal structures like the actin, intermediate, and microtubules also get rearranged. So the actin filaments, which are initially scattered throughout the cytoplasm, you can see that when the nucleus is going to break down, that is the GB is going to break down, it starts getting arranged in the periphery. And in mature oocytes, they're completely arranged in the periphery. Intermediate filaments, which are usually present in the corners, actually start getting arranged, scattered evenly through the cytoplasm, which is going to maintain the strength. And the microtubules also, first during the GV breakdown stage, they come towards the nucleus, which help in the nuclear membrane breakdown and the further meiosis. And it is present just below the polar body in a mature oocyte. So this is very similar to what I said. So you can see this is a better picture which shows Golgi apparatus and all that being a solid structure which starts breaking down and gets evenly distributed. The endoplasmic reticulum, the Golgi apparatus and all those cells and the cortical granules. So in this picture, you can clearly see how the cortical granules are in the periphery and how the actin is also in the periphery. The microtubules are under the first polar body, and the intermediate filaments are scattered in the cytoplasm. So I've shown the picture 3 times, so I hope it's strongly registered in everyone's mind. So now we've spoken about oocyte maturation. So what happens during fertilization? So to briefly tell about the steps of fertilization, once the sperm is capacitated, it comes and binds to the zona, and then the acrosome reaction takes place, and then the sperm and the oocyte plasma membrane fuse. So as soon as the fusion happens, the oocyte activating factor, which is the phospholipase C zeta, enters into the cytoplasm. And this produces inositol triphosphate and diacylglycerol, which in turn activates protein kinase C. And the inositol triphosphate acts on the endoplasmic reticulum and releases the calcium in an oscillatory manner, which helps in oocyte activation. So just as when all these are happening simultaneously, the cortical granules, which are present in the periphery, like what we saw in the previous images, are going to release their contents into the perivitline space and thereby harden the zona in order to prevent polyspermy. So these functions, these multiple functions, for all these functions to happen, we require the effect of cytoskeleton. So cortical reaction, like I said, to release it, we need actin plays a major role in this one. In oocyte activation, that is basically for rearranging the organelles and for releasing the contents from the endoplasmic reticulum, we need cytoskeleton. And for spindle reorganization, the microtubules play a major role. So my majority of my talk is done. I just want to highlight certain terminologies that are more commonly spoken about, and most of it I've already mentioned about in the presentation. And I think to help understand the next topic better, if you know this terminology, then maybe we can interpret more from it. So what are centrioles? Centrioles are paired barrel-shaped organelles located in the cytoplasm near the nuclear envelope. They play a role in organizing microtubules that serve as a cell skeletal system. And they also determine the locations of the nucleus and organelles within the cells. There is something called pericentriolar material or pericentriolar matrix, which is basically the closest proximity of the cytoplasm around the centrioles. And there is another term which is called MTOC. If you see a lot of IVM articles, you'll read about this MTOC, which is a cellular structure frequently assembled on the surface of an organelle. It organizes the microtubules by controlling the nucleation and stabilizing and anchoring them, which is also called as the centrosome. And also there is a term which is called phosphorylation and dephosphorylation, which is very similar to polymerization. It basically means that there is an addition of phosphate group to a molecule, and dephosphorylation means removal of phosphate group from a molecule. So I had already mentioned about this one, where the centrioles are actually absent in the oocyte of a mature oocyte, and it has to come only from the sperm. So when that doesn't happen properly also, there'll be problems. I think the next speaker will speak about that. And disruption of cytoskeletal function, I'm not going into detail. So I think with all the things that I have spoken so far, we definitely know that if there is any problem with the cytoskeleton, then we are going to have problems with oogenesis, with fertilization, and further embryo development. So to summarize, the cytoskeleton, it basically holds everything together, and it is absolutely essential to keep the oocyte together. The actin, intermediate filaments, and microtubules are the 3 major components of the cytoskeleton, and it has varied roles in an oocyte. It helps in maturation, fertilization, and post-fertilization changes, and also for the further embryo development. So the cell and I must say thank you, because this actually provoked me to read a lot and to revise all my basics. So I once again thank you all for this opportunity.
Thank you so much, Dr. Sindhuja. It was a wonderful lecture. It was exceptionally well articulated, and I believe that all will agree that you clarified many fundamental yet crucial points in the presentation. Do we have any questions for Dr. Sindhuja?
I think, Nancy, can you check that chat box? Is
Sir,
there any question?
because the chat box is not connected with the audience, we don't have audience question here.
Acha, okay.
Yes,
So,
wait, sir, I couldn't see any question from in chat
so
box.
there is one question from my side. Sindhuja, it was an excellent lecture actually. And can you please, there was one slide you mentioned that when you hold the oocyte, you have to take care about that, how you holding that oocyte cytoplasm. Sometimes it is, you know, that pressure is too much and you are holding that oocyte, it will damage the oocyte. So can you tell us about how, at which extent you can aspirate the oocyte? Is there any way how we can see that cytoplasm does not go inside that too much inside the holding pipette? Can you explain that thing little bit?
Yes, sir.
During XC.
Yes, sir. So, so ideally, if all the microtools and the gametes are in the right plane, then there will not be a situation where we need to have too much pressure to hold the oocyte. So when we are in the exact focus of the oocyte and the holding pipette, then we can make sure that actually so much pressure is not required to hold the oocyte. Just the zona will be inside the holding pipette, and that pressure is enough. But in situations where there is a sort of even a small angle change, then we will be actually using too much pressure to hold the oocyte and in order to rotate the oocyte, and that's when the situation happens, sir. I hope.
So sometimes I think sometimes what happens when you are holding the oocyte and you are not able to remove full cumulus, then that kind problem can occur at that time also.
Yes.
That is the thing. Thank you, Sindhuja. It was wonderful lecture. Thank you so
sir.
much.
Thank you for the opportunity, sir.
Thank you, Dr. Sindhuja, once again for this.
Well, can I interject? I have one question for Sindhuja. Hi, Sindhuja. It was a nice presentation.
Speaking. Oh, hi, Sanket. How are you?
I'm good. And for the sake of the younger audience, could you please explain? Of course, as you rightly mentioned in your talk, whenever we do ICSI, which is not supposed to happen in nature, we tend to break certain microtubules, right? Which are part of cytoskeleton. So how does this going to come back? Polymerization or depolymerization? Because we tend to aspirate the oocyte as well, right? So could you please explain that how it's going to reform?
So like I said, Sanket, all the organelles in a mature oocyte are arranged in a way that it is going to keep the oocyte very stable. So when we aspirate a few nanoliters, I won't even say microliters, we're aspirating very few nanoliters. The only organelle that actually gets disturbed probably is the mitochondria. And even that does not have a
endoplasmic reticulum as well. Because
of course.
The center.
Yeah. So like I said, it is not, that's why we don't go inside a smooth endoplasmic reticulum and, pierce it, because there it is present as a cluster. Otherwise, all the organelles are evenly distributed in the cytoplasm. So we're not going to do damage to it when we are injecting. But like I said, maintaining the temperature and maintaining the osmolality of the media and the culture environment is absolutely essential if we want to make sure that these organelles are functioning in the right way.
Right. So the point is yes, of course, we do them. Sir?
Can I just add up to what Sindhuja said?
Please, sir.
Please, sir.
See, the simplest thing is that whenever you perform this one, she said nanoliters, but unfortunately, we will not be sucking in nanoliters. We will be sucking in picoliters.
I'm sorry. Yes.
10 to the power of 12 is the amount.
Correct, sir.
This polymerization and depolymerization is a continuous process within a cytoskeletal operation. So whenever you disturb a microfilament, other than the endoplasmic clusters and other things, obviously, if it is a smooth endoplasmic reticulum, you will see it as a cluster. But other organelles and other microtubules they are all submicroscopic. In our ICSI level microscopy, you will not be noticing it. So we will be inducing depolymerization. And the moment you inject the sperm back and the entire cytoplasm back, the
There's a carriage.
repolymerization also
Also
starts.
happens. Yeah.
It happens, and it has to happen. Whenever you have a trouble in repolymerization, those oocytes are the oocytes which go into necrosis.
Yes.
Mm. Correct. Because the you're breaking the very base of the integrity of the cell, so
Yes.
that they won't withstand the physical stress that we are gonna incur. So I mean, that's the whole, as she rightly said in the beginning of the slide, it's the base of the structure of the cytoplasm, right? So
Yes.
that supports the integrity of the cell.
Sinduja, congratulations. You presented it really well. Nobody
Thank you, sir.
understands the
Means a
role
lot.
of actins and other things, microtubules, because the moment we say cytoskeleton, everybody will sit with only endoplasmic reticulum and Golgi apparatus. You definitely talked about spindle and all other microtubules. It was a fantastic compilation. Very good.
Thank you, sir. Thank you, sir.
Yeah, over to the coordinators.
I think we are ready to move towards our next lecture, which is going to be presented by Dr. Charu Jandel. Now we are going to talk about micronutrients and how they play a crucial role in improving the oocyte quality. For that, we have with us Dr. Charu, and she is consultant super specialist, Department of Reproductive Medicine, ESIC Medical College, and she is ex-consultant Nova Southern Fertility and IVF, New Delhi. She is also professional member of ISAR, IFS, and many other organizations. She has published more than 15 publications, research papers in indexed journals. Now over to Dr. Charu Jandel. Please join us.
Yeah, thank you, Nancy. Good evening, everyone. Thank you, Ahira, for having me here and giving me this platform to speak. Today, my talk is the role of micronutrients in improving the oocyte quality. Can I please share my screen? Is that okay?
Yeah, you're good to go, man.
Yeah, thank you. So, the journey towards parenthood can be a tiring one, apart from the financial burdens and emotional ups and downs. So, the causes of fertility are diverse and often multifactorial, we all know. And there are various factors which are ovulatory dysfunctions, tubal and pelvic pathology, sperm abnormalities, as well as the unexplained factors. The lifestyle changes, weight, stress, smoking, and alcohol consumption can also significantly influence the conception. While the influence of minerals on male fertility has been extensively studied, their role in female reproductive system has been marginally addressed. Diets high in trans fats, refined carbohydrates, and added sugars can add negative effect on fertility, the literature suggests. Conversely, a diet based on Mediterranean dietary patterns, for example, rich in dietary fiber, omega-3 fatty acids, plant-based proteins, vitamins, and minerals, has a positive impact on female fertility. So micronutrients won't be wrong to suggest that they are crucial, but unfortunately overlooked elements. What are micronutrients? Micronutrients are essential vitamins and minerals that are required in very small quantities as dietary components. Although these micronutrients do not provide energy to the human body, they are essential for various enzymatic activities and the metabolic activities occurring in the body. Micronutrients play a multifaceted role. They have a role in improving the oocyte quality. They play their role in ovulation, embryo development, oxidative stress, and miscarriages can be because of the poor oocyte quality. This is a picture which shows that the diet rich in high PUFA, multivitamins, minerals has genetic and epigenetic modifications and has a role in gut microbiota, and therefore success rate in fertility in natural conception as well as in the ART success go up as they improve the oocyte quality, blastocyst formation rate, embryo implantation rates are higher, leading to high live birth rates. On the contrary, the animal-based proteins rich in carbohydrates, sugars, trans fatty acids, and Western-like diet have a lower ART success. So these are all modifiable risk factors. What do we need to do? Supplementation before conception can help in restoring the micronutrient status to the recommended levels and reduce oxidative stress when antioxidants are included. Especially, it's vital in the context of infertility caused by inflammatory conditions like endometriosis and ovulatory disorders like PCO. This is the slide showing what is oxidative stress. Inside the human body, as the cell metabolism happens, the reactive oxygen species are released as a process of normal metabolism. Normally, these reactive oxygen species help in various enzymatic activities, leading to various functions in the body. And normally, antioxidants are produced by the cell's defense mechanism, which keep these in check. Problem happens when these reactive oxygen species increase than the defense mechanism of antioxidants and the state of oxidative stress is produced. The second diagram that shows the blue picture that shows when there are less oxidants and there are more antioxidants, this is a state of health, that is oxidative eustress. And the other one, which is the red part, shows more oxidants and less antioxidants, that is the oxidative stress. And this is the state of illness. In this picture that shows antioxidants are less in number and oxidants or the reactive oxygen species are more in number, leading to oxidative stress. So what does this oxidative stress lead to? It causes cell damage in the form of lipid peroxidation, DNA damage, it leads to protein and enzyme oxidation. It also leads to the poor spindle formation, errors in the spindle formation leading to aneuploidies, also leads to telomere shortening and DNA damage. Although ROS are necessary for normal cell functions, excessive ROS production can happen with smoking, alcohol use, excessive body weight, exposure to environmental toxins, advanced maternal age. These all can impair physiological female reactions like oocyte maturation, ovulation, luteolysis, and follicular atresia. These all can lead to infertility and especially in the inflammatory conditions. So apart from the intrinsic and the extrinsic factors, gamete manipulation can occur during the ART procedures, like exposure to external stressors or other environmental factors. Despite the recent advances in IVF settings, it does not recreate the condition of natural fertilization, which includes tight physiological regulation of oxidative stress by antioxidants. So oxygen concentration, temperature variation, high light exposure, culture media composition, and cryopreservation methods are environmental sources of oxidative stress in the IVF lab that coincides with a decline in fertility and an increase in embryo aneuploidies. Human IVF culture media are supplemented with combinations of molecules with antioxidant properties, including human serum albumin, ETA, folic acid, ascorbic acid, and pantothenic acid. Antioxidants are endogenous to organisms, but it is uncertain if the supplementation of these substances can improve oocyte mitochondrial functions. So what basically antioxidant is? It is a substance that delays the oxidation of lipids, carbohydrates, proteins, or DNA by any of the three methods, directly scavenging ROS or indirectly upregulating antioxidant defenses or inhibiting ROS production. On one hand, lipid peroxidation affects the integrity of cell membranes, while it also causes nuclear DNA degradation, inducing the onset of apoptosis. Antioxidants can be supplied by oral supplementation, which can be given in the form of prenatals or before starting an IVF cycle or along with the stimulation cycle, while the culture media supplementation attempts to do this in vitro. This is a list of antioxidants which shows if there is any proven benefit in oocyte or embryo quality, and this shows CoQ10, resveratrol, melatonin, vitamin B, and vitamin E have a proven role in improving the oocyte quality. Vitamin A stands a question. So CoQ10 carries electrons in the mitochondrial respiratory chain and participates in the synthesis of ATP. While the levels of CoQ10 decrease with the advancing age, this decline coincides with a decline in fertility and hence an increase in the embryo aneuploidies. In humans, it has been studied the higher CoQ10 levels in the follicular fluid are related to improved embryo quality and high pregnancy rates. Another paper published in 2018 suggested that CoQ supplementation given in a dose of 600 milligrams per day for 2 months before ovulation stimulation increased ovarian response, that is 4 versus 2 oocytes, and the number of high quality embryos. This is in patients with a poor ovarian reserve. However, no difference in clinical pregnancy rate, miscarriage, or live birth rates were observed in this study. Another study of Reproductive Biomedicine Online 2014 suggests that CoQ10 supplementation benefited PCO pregnancy patients who exhibited ovulatory disorders and higher proportion of immature follicles within the ovary. Coming to resveratrol, this is a natural polyphenol synthesized by several plants in response to pathogens. This is found in grapes, red vines, peanuts, and several medicinal plants. It has anti-aging and antioxidant properties, anti-inflammatory also, and it has insulin sensitizing and cardioprotective roles also. In the reproductive field, resveratrol may benefit women with diminished ovarian reserves, PCO, endometriosis, and uterine fibroids. This may improve age-related decline in ovarian functions through the activation of SIRT1 molecules. However, let me mention that we have no experience with this molecule. Melatonin is another one such molecule which is found high in mitochondria, and it has an important role in reducing the oxidative stress. Its antioxidant properties come from its excellent free radical scavenging, as well as its capacity to upregulate the expression of antioxidant enzymes and a spectrum of stress-responsive genes. In addition, melatonin indirectly accelerates the electron transports, decreasing the electron leakage and reducing ROS formation, and this process is called radical avoidance. Melatonin levels again in follicular fluid are suggested as a biomarker to predict IVF outcomes and ovarian reserve. Its production decreases in age, and in the ovary, this age-related decline is related to increase in FSH hormones associated with menopause. Therefore, this may delay ovarian aging. Vitamin A is found in green and yellow vegetables, dairy products, fish, egg, and organ meats, and its metabolites have important roles in all the steroidogenesis and follicular growth, oocyte maturation, and embryo development. Folic acid, again, is an age-tested micronutrient, and it is a methyl donor to support the methylation of homocysteine to become methionine, which is critical intermediary in the production of glutathione, which is the primary intracellular antioxidant. Apoptosis is essential for regulation of follicular atresia, degeneration of corpus luteum, and endometrial shedding, and this is influenced by folate and zinc. Hence, they are an important constituent. The risk of luteal phase deficiency may be increased in women with inadequate selenium levels. Ascorbic acid, again, supplementation is significantly increased in serum and follicular fluid levels. However, no difference in implantation or clinical pregnancy rates were found after ascorbic acid supplementation in women undergoing an IVF procedure, either during hormonal ovarian stimulation or during the luteal phase. Although promising, vitamin C supplementation in fertile patients has yet to show a beneficial effect on the fertility. Follicular fluid vitamin E levels ranges between 0.35 to 1 milligram DL, and this is related to higher oocyte maturation rates, while serum vitamin E levels in the range of 10 to 15 milligrams per deciliter were related to higher proportion of high-quality embryos. Again, no such relationship between vitamin E levels and the oocyte maturation was found, nor was there any correlation between follicular fluid vitamin E levels and the embryo quality. Omega-3 fatty acids from oily fish or supplements have a beneficial effect on growth and maturation of oocytes. It decreases the risk of anovulation and improves embryo morphology and are associated with a higher concentration of progesterone. However, the results of association between omega-3 fatty acids and fertility are contradictory. In numerous studies, no or insufficient evidence has been observed. Let's have a word about the dairy derivative fats. The results are again often contradictory. On one hand, according to the study, the consumption of low-fat dairy, including low-fat milk, yogurt, cottage cheese, increased the risk of infertility because of anovulation, while high-fat dairy increased fertility. This may be possibly associated with a high content of estrogen and fat-soluble vitamins in high-fat dairy. Presence of trans-palmitoic acid, which seems to improve insulin sensitivity. On the other hand, another researcher did not confirm that the consumption of high-fat dairy is correlated with increased fecundity, and they did confirm that the consuming lactose and low-fat dairy did not negatively affect fertility. Animal protein consumption has been associated with a higher risk of infertility due to lack of ovulation. The difference may stem from the impact of plant and animal protein on insulin and IGF-1 secretion. Insulin response is lower after plant protein consumption than following the animal protein. It has been reported that low serum concentrations of zinc and selenium were associated with a longer period for achieving pregnancy. Additionally, a deficiency of selenium and copper, but not zinc, was linked to a high risk of infertility. Selenium possibly influences the growth and maturation of oocytes. Again, magnesium also takes a part in glucose metabolism and has an effect on oocyte metabolism. So, there are no clear guidelines on supplementation to enhance fertility in women. And the bottom line remains a properly balanced diet should provide all minerals and vitamins except for vitamin D and folic acid, which should be supplemented. This is a very interesting study that shows iron status in women with infertility. 72 participants were there in the study, and this shows that ferritin levels below 30 micrograms per liter was found in patients with unexplained infertility. So, medium range of serum ferritin has to be there for a good oocyte and a good fertility levels. Again, it has shown that a higher ferritin levels were associated with infertility. Selenium, zinc, and copper are essential for maintaining reproductive health, while exposure to toxic metals like cadmium and lead is detrimental. So what does Cochrane say? It says we are uncertain whether the use of antioxidants will increase live births, as evidence was of very low quality. Based on our results, we would expect that out of hundreds of fertile women not taking antioxidants, 20 would have a baby, compared with between 24 and 36 women per 100 who would have a baby if taking antioxidants. Low quality evidence suggests that antioxidants may be associated with increased clinical pregnancy rates. So the current evidence is insufficient to recommend nutrient supplementation to improve female fertility due to low level of evidence. However, there is currently no indication that these nutrients pose any risk or threat or any significant harm. There is a need for high-quality RCTs to identify both the efficacy and safety of nutrient supplements for female fertility in women trying to conceive naturally and also in women using medically assisted reproduction. Thank you for a very patient listening. Thank you.
Thank you so much, Dr. Charu. It was a wonderful presentation. But do you have a question or can I ask?
You can ask, Nancy.
We don't have any question in the chat group, but only one thing which I want to ask, if you have to compile everything and you have to give a few lines take home to the students who are listening to our webinar, what should be the 2-3 line compilation of the presentation?
Yes, first we can zero in on those that subgroup of patients which are high risk, like advanced age, those with inflammatory conditions like PCO or endometriosis. We can zero in on those patients, plus those patients who are who have a low reserve of these micronutrients, like in sort of anemia or low socioeconomic strata, where we want to supplement them with the micronutrients. We know the dietary levels are not that up to the mark. In those patients, we can supplement them with the micronutrients. And the micronutrients which have a role, which have a prominent or the upcoming role is copper, zinc, selenium, and CoQ 10, of course.
Okay, thank you so much, ma'am.
Dr. Charu,
Sir.
One question. So how you decide that, what are the parameters you decide that you have to start with that micronutrient for this patient?
So like advanced maternal age is one of the factors in patients with PCO, in patients with endometriosis, lower socioeconomic strata, where we see the patient is of poor nutritional status, In those patients, we can definitely start.
Do you have an idea about the melatonin? Is there any
Yes.
role of that melatonin in this?
Yes, again, it is controversial. One of them is that another role is that melatonin increases the mitochondrial capacity. Mitochondrial function is improved and induces a good sleep, and a good sleep also induces a good fertility outcome.
Thank you, Dr. Charu. It was a wonderful lecture, and I hope that,
Dr. Charu, can I ask you one question?
Ma'am.
It was a wonderful talk. I just wanted to ask, do you think measuring these micronutrients would be the next future? I mean, wherein if you have a high-risk patient, you start measuring these micronutrients and
Maybe,
then
yes.
you can treat them accordingly?
Yes,
is there
maybe.
any way that we can measure these micronutrients? Do you do it?
Yes, micronutrients can definitely be measured. There are recommended daily allowances for these micronutrients. Like for selenium, it is 55 micrograms. For zinc, it is 8 milligrams. And vitamin A, D, E, and K, we all know. And again, copper is 900 micrograms and magnesium is 300 milligrams. So maybe in near future, we will be able to measure and we'll focus on that.
Probably do a focused
Yes.
treatment for the high-risk patients.
Yes.
Okay, thank you.
Can I make a comment here?
Yes, sir.
Apart from all these scientific jargons and all those things, I firmly believe that all those things, very fancy sounding like the Mediterranean diet and all, it is the physical fitness and activity that matters more than the diet. It is my firm belief, because we have been reproducing for centuries, all parts of world, and they are like our grandparents. They were eating what they were eating, but still they had less infertility problems. That, in fact, we had over fertility problems. So unfortunately, we gave up the physical activity and focused on the diet only. So that is the problem. And if we, as infertility professionals or the healthcare professionals, focus our, measures to improve the quality of life by introducing the physical activity in our lives, we can achieve more. And I'm firmly believe that many of the infertilities can be tackled without any infertility treatment interventions.
Nancy, where are you?
I'm here only, sir.
Why everyone is stunned?
Yeah, that's why I'm telling where is Nancy.
I'm here only, sir. Can we move further? Thank you so much, Dr. Charu. It was a wonderful presentation, and we took a few beautiful points from both the last presentation. Now, moving towards our next lecture, our next guest speaker is Dr. Juan J. Zamora. He is going to speak on cytoskeleton abnormalities and their impact during IVF and ICSI. Just a second. Dr. Juan J. Zamora is a PhD physiology from Barcelona, Spain, and he is a scientist in reproductive medicine. And he works in R&D Eugene Group also, and he's also ASHRAE JC editor. Dr. Zamora, the stage is all yours to present. Welcome.
Before starting that lecture, I would like to thank you, Dr. Juan. Actually, I tried to call 2-3 people for that lecture, but nobody accepted this lecture. And Juan is one who accepted this lecture with very enthusiasm, because he thought that lecture will be very good and it will be benefited for all. So thank you so much, Juan, for joining us. Thank you.
Okay, no, thank you. Thanks for the invitation. I actually I see the difficulty for many people to accept the talk because I think you picked the topic very, very properly because this topic is the bridge between the clinical and basic research knowledge. So since I think the advantage was that I spent almost 15 years doing only basic research. So I had a little bit of the knowledge to try to connect these things. And let's see, I will just tell you the story that I found by looking at references and the dots I can connect based on knowing a little bit of clinic and a little bit of basic research. So let me just find my presentation here. There you go. Do you see it? Okay. So there you go. So basically, the presentation is cytoskeletal abnormalities and their impact during IVF and ICSI. So we usually look at the oocyte using these wonderful images of Hoffman microscopy that are very useful for the IVF lab, that gives this kind of three-dimensional idea, so we can recognize polar bodies, the zona pellucida, and other structures inside. But since we are so used to these images, we sometimes forget that underneath these images or underneath what we see, there are molecular and cellular structures that actually give as a result a nice morphology to the embryo and also a nice health to the embryo, meaning that these oocytes that are healthy or we can say that they have good quality can go on and be fertilized. And this is one of the things that we usually miss when we talk about good morphology. We assume this is good quality, but it's not always the same, right? Good morphology doesn't really mean that the whole embryo is healthy. We can try to assume that, but not always. And we will see a little bit why or how. For example, there are two things that give the shape and the morphology to the oocytes. One is the actin cytoskeleton. This is a dynamic network of actin polymers and actin binding molecules that regulate the cell structure, shape, and also organelle transport. So I'll try to give some examples on what does this mean. And also the microtubule cytoskeleton. So the actin cytoskeleton will be this here in magenta here. And the microtubule cytoskeleton, which is a dynamic network of tubulin bundles that facilitate also cell division, because as you can see here, this is the spindle of the oocyte. It also helps transport cellular materials and organs. So this is very important, not only for the structure, but also to reorganize the whole organelles or materials inside the cell. Okay, so this talk is going to be divided into three topics, or three parts, I mean. The first one is going to be the oocyte cytoskeleton and its association to oocyte quality, or what we would recognize as oocyte health. The second one is going to be a bit shorter, but it's going to be on biomechanical characteristics of oocytes that come from all these cytoskeletal structures. And the last one is going to be on something that we haven't explored in the field, but there's an example using non-model organisms, and it's called the cytoskeleton and oocyte wound healing. What happens when we make a wound in the oocyte and how does it heal? Okay, so let's start from the beginning. So what I wanted to show you first is a little bit of a history on oocyte morphology, how it has been used in the field. I found, I didn't go too much into detail about this, but I found this paper from 1999 in which there was already an observation saying that oocyte morphology correlates with embryo quality and pregnancy rate. So this suggests that we were already looking at nice, pretty oocytes, and more likely they were giving rise to embryos that had good quality and could result in a pregnancy. All this information led later on to propose the different characteristics, morphologies of different oocytes, and see whether these actually give rise to worse embryos and prevent pregnancy when you were injecting and transferring these embryos. So most of these characteristics were observed in the lab. Like, for example, from these pictures, you can see granules in the cytoplasm, a big disc of endoplasmic reticulum in the center, some of these granules around here, very big, like you can see here, or vacuoles that look like structures with something inside. We don't really know what these were. Or we could also see malformations in the zona pellucida, or a very big perivitellin space with debris inside it. All these were considered malformation or dimorphisms that were not giving, as a result, a good embryo that could result in a pregnancy. And actually, there's a lot of, well, not a lot of literature, but there's some literature trying to mix the oocyte quality or oocyte morphology, I should say, and the results in the outcomes from an IVF treatment or an ICSI treatment. The thing is that most of the work is contradictory. Some say yes, it is very important, and some others say no. So the only thing I'm going to present about this today is kind of the latest systematic review that I found from Nikiforov in 2021, which says that these authors have revised the whole literature, and they say that there's no influence if you find in your oocytes a dark color of the cytoplasm, which is in an incidence of 7% of most of the cycles. And there's no influence if you find homogeneous granularity of the cytoplasm, something like this oocyte here, which is actually more common in 19% of the oocytes they get. And also there's no difference, at least if you inject ovoid shape of oocytes. It seems that the shape doesn't really matter. They can give rise to nice embryos. I have to say here that the outcome is embryo formation. It's not live birth or healthy baby. So we don't know how far we can make conclusions on this. But at least if you find this kind of oocytes, there's no influence in the outcome. So you can still form blastocysts. The characteristics that will likely affect the treatment outcome are abnormalities such as refractile bodies, which are some of, these structures here, or smaller ones. This is also another thing that is difficult to define what a refractile body is, because they depend on the type of microscopy that you're using. But also something that you can score easily is if you find a fragmented first polar body. This is likely affecting the treatment outcome. And actually, this is very, very common. If you find a dark zona pellucida, can be bad. But also if you have enlarged perivitellin space and debris in it, like this picture here. So this is also not very good. It can affect the outcome. It can affect how many blastocysts you can get. And I mean a good morphology blastocysts. What definitely affects the outcome negatively is cytoplasmic vacuoles. So like these very big structures here. And centrally located cytoplasmic granularity, which is in this picture here in B, which correlates usually also with clusters of smooth endoplasmic reticulum. Okay. So these are the things that definitely affect the outcome, likely affect the outcome, and no influence. But as I said, we're still far from knowing what does affecting the outcome mean, because it can be just making blastocysts, but we don't know yet if it also means getting a healthy pregnancy and also a healthy live birth and a healthy baby. Okay, so the most important thing is that we don't know what does this mean to the health of the embryo, because we don't really know what these structures correlate with at the cellular and the molecular level. We're making connections and we're working on that as a field. I mean, we can find the evidence on this, but the main problem is that for some reason, we're not making the final connections to try to find and explain what is it that these structures give to morphology. So what I'm going to do here right now is I give a brief history of oocyte morphology or quality and the cytoskeleton. So the earliest paper I found about cytoskeletal organization in oocytes is this from 1988, in which you can see that the authors explain how the actin cytoskeleton and microtubule cytoskeleton start forming when the oocyte is maturing, and how these are very important structures that they already observed in 1988 that have to be there to give a healthy oocyte during maturation. We had to wait a long time until 2013 to find more specific cellular and molecular structures. For example, of course, there were other articles, there were more discoveries, but especially this group from Melina Xu in Germany started asking the main questions on what are the structures, molecular and cellular structures that can give rise to bad oocytes and that can relate to morphology. And for example, in this paper, Holubkova et al. Found that vesicles, these small vesicles here that you can see circles here, modulate an active network of actin that allow for asymmetric spindle position. So the vesicles that we see here make that the spindle go to one of the sides so a polar body can form, right? So you can imagine if these vesicles are not healthy and they start aggregating, probably that's going to lead to a wrong spindle formation, asymmetric positioning. Some other people also work on oocyte maturation, and the result was, a more detailed a description on what happens during oocyte maturation. For example, when you have a GV, the oocyte is completely immature. You have microtubules, actin bundles, but nothing very well defined. As the GV starts maturing, the oocyte, the nucleus starts maturing too, but also the cytoplasm starts maturing. And what does this cytoplasmic maturation mean? We don't know yet. What I suggest is the formation of cellular structures like the actomyosin cytoskeleton around the cell, the movement of specific organelles around, the processing of the endoplasmic reticulum to reduce it in specific forms for the oocyte, the formation of granules around the oocyte, and also the movement of mitochondria. Of course, the formation of the spindle, and then you get a mature M2 oocyte that is ready to get fertilized. So oocyte maturation involves the movement of a lot of different organelles and the formation of the actin cytoskeleton as well as the microtubule cytoskeleton that forms the spindle. Later on, also, CoQ et al. Found that for an oocyte that is maturing, it is very, very important that you form an actin cap here. So you can see here the actin cytoskeleton surrounding the egg, and you see the microtubule one with the spindle. But this actin cap is going to help this oocyte to extrude the polar body. If you don't have an actin cap here that forms, your polar body is not going to extrude properly, and then you're going to end up with oocytes that are not going to fertilize properly. And this could be linked also to this result I show you on fragmentation of the polar body, which is something we observe on light microscopy images, but might have, and I'm sure it has this cellular and molecular explanation. And another thing that I found here is this is the only paper that links actomyosin cytoskeleton and dysmorphic patterns in human oocytes. So Dalcanto et al. In 2017 published in Human Reproduction that dysmorphic patterns in oocyte morphology are associated with cytoskeletal alterations. And you can see here that a normal oocyte, that is a control oocyte, has a continuous structure of actomyosin, a nice spindle, and also a nice pinched polar body. But dysmorphic oocytes, they have a broken actomyosin cytoskeleton and very strange spindles form and not a very good polar body, right? So in this case, I think from what I found, this is one of the very few, if not only papers that kind of make this link between the actomyosin cytoskeleton and the morphology grade of oocytes. And as I explained you before, this is not only about the oocyte morphology, but it's also about the positioning of all the organelles, because these vesicles here that we can see in the oocytes help make this actomyosin structure that help position organelles and the spindle in the right place at the right time. So 2 years ago, 2023, this group, Tatikova et al., using ultrastructural techniques, they explain how human oocytes cytoplasmic abnormalities link to the cytoskeleton dysfunction. And basically, I'm not going to show the whole papers here, but this is the nice summary of the whole figures that they have. You have a normal human oocyte that matures, as I explained you before. The nucleus matures, forms a spindle, and also a polar body. But also the organelles start reorganizing, forming specific structures that are going to be useful for when fertilization occurs. But when you have cytoplasmic abnormalities, like cytoplasmic granularity, these result from an aggregation of organelles or structures within this oocyte. Or smooth endoplasmic reticulum discs that you can see here is the accumulation of the endoplasmic reticulum for some reason. We don't know why, but they are accumulating. If we go and look for other cell types, we know that these things occur when the cells are under stress. And specifically, stress that produces oxidative stress or aging or whatever reason. But we still haven't found this link between the cytoskeleton, organelle abnormalities. We kind of know them, right? You saw in the previous talks about oxidative stress, what is going on, but we haven't made this link in which we know exactly what's going on at the cellular and molecular level. Another thing is vacuolization here, or refractory body, which is basically a lysosome that is being formed in the center of the oocyte. And lysosomes form usually when there's a lot of stress and the cells need to get rid of a lot of misfolded proteins or molecules that are toxic for the cell. The main interesting thing here is that this author perturbed the cytoskeleton with drugs that get rid of the actomyosin cytoskeleton, as you can see here, and also with drugs that prevent the formation of the microtubule cytoskeleton. And most of these treatments also result in similar structures that are observed in normal eggs with dysmorphic patterns. Now, like, for example, this cytoplasmic granularity, if you get rid of the actomyosin cytoskeleton with cytokalasin B, you see that the shape of the oocyte is very bad. So very large perivitellin spaces, and you see all these aggregations of cytoplasmic granularity here, which are basically organs that are not being positioned properly. If you also use Brefeldin A, similar thing happen. You still see a nice form oocyte, but with a lot of this aggregation of basically endoplasmic reticulum and atypical mitochondria. And if you use nocodazole or paclitaxel, which get rid of the microtubules, the spindle doesn't form. Also, the polar body doesn't form. You can see here, and you have aggregations that start forming some refractile bodies, which are lysosomes. So that means that obviously these eggs are sensing this toxic environment and are producing a lot of molecules that are toxic for it. So basically, we are, as I said before, we're very close to know what these structures are. We still haven't found the direct link. I think this is something we should work on as a field in trying to find these dysmorphic patterns. What do they correlate with exactly from cellular and molecular structures? And how is it that we can prevent them, right? How can we prevent this? And if they really form bad blastocysts and prevent the reaching a pregnancy. Or maybe they form nice blastocysts, but that don't end up in a pregnancy or end up or don't end up in a light burn. Another interesting thing is that instead of going to that understanding at the molecular and cellular level, what we're doing is using machine learning or artificial intelligence algorithms that can give us a score on how likely these oocytes are going to give rise to competent blastocysts. And nowadays, luckily, some of these algorithms are telling you what are the features that are most important for positive outcomes or negative outcomes in oocytes that are competent or incompetent, right? And interestingly, you can see that in oocytes that are not dysmorphic or somehow dysmorphic, for example here, the important features are those related to cell surface, right? For both competent and incompetent, and for some aggregation of structures here. So, I think maybe it's just that I am a little bit biased, but I think we should really focus on understanding what are the important features that can tell us what a healthy a healthy oocyte is. And I think this is going to be related to the function of the actomyosin cytoskeleton and how this is working during maturation in order to localize properly the organelles that are inside the oocyte. So probably this information is more important than just using artificial intelligence. We need to use these methods in order to take better decisions in the clinic. But in order to really understand and trust these methods, we really need to make this link with basic research. And specifically because, if these are the features that are important and they relate to a healthy actomyosin cytoskeleton and a healthy cytoplasm, then we can exactly know that these dysmorphic oocytes are not going to make it to blastocyst stage or to give us a pregnancy. So what are the molecular and cellular features that correlate with good morphology and digital image information and the actual oocyte quality? Okay, so now I'm going to go a couple of slides into the biochemical characteristics of oocytes. And at the end of this couple of slides, I will make the connection with the actomyosin cytoskeleton. So this is just going to be based in one single paper because not much has been done about this in the field. But this is a paper published this is not Human Reproduction. I think this was Nature Communications. So this is a mistake. But what these authors did, Yanes et al., they took advantage of biomechanical tools to measure the mechanical characteristics of the oocytes. What does this mean? If you have an oocyte here, and you go with an aspiration tip, you can aspirate the plasma membrane of the oocyte together with the zona pellucida, as you always do, for example, for doing an ICSI. So these are tools that actually we have in the embryology lab. It's just that we never use them for this purpose, right? And you can aspirate, and you can see here that as you aspirate, there's going to be a depth of this part of the cell that is going to go inside, and you can measure that. You can relate it to the pressure that you're applying, and you're going to see how much by applying a mathematical model here that I'm not going to go into detail, but you can get the viscoelastic properties of this oocyte. Viscoelastic means how stiff or how soft this tissue is. The interesting thing, though, is that they measure this in oocytes right after fertilization, and then they follow whether they form a viable or a non-viable blastocyst. And you can see that non-viable blastocysts, shown here in blue, had a larger distribution of the mechanical parameters, while the embryos that were viable, that resulted from these eggs, have a less dispersed distribution. And they calculated these biomechanical parameters from this model. And the interesting thing is that when they plotted all these biomechanical parameters, you can see that the blastocysts that were viable concentrated around here, the center, but the ones that didn't give rise to a viable blastocyst were more in the external part. And these blastocysts, when transferred, The non-viable ones resulted in less live births compared to the viable ones that resulted in more live births. So this, to me, it just shows that by knowing a little bit more about the biomechanical parameters in the oocyte, we can learn a little bit better to discriminate which are the ones that are healthy ones. The only problem is that for this, we need people, engineers that can help us and make these measurements. But we have the tools in the lab, and I mean in the IVF lab. So we need more interdisciplinary collaborations for this. What these people found as a summary here is that you have an immature oocyte that undergoes different parts of imaging damage or external stress, say cell cycle arrest, they express genes. And based on this, the stiffness of the oocyte is going to change. If you have a very stiff oocyte, is less mature, and this is usually not going to give rise to a good fertilized embryo. If you have a low stiffness oocyte, it can two things can happen after they get fertilized. It can be viable if it's low stiffness, but if it's too soft, it can also result in a wrong blastocyst or pregnancy. The interesting thing here is that these biomechanical parameters, it has been already known for a long time that they relate to the actomyosin contractility, so to the actomyosin cytoskeleton. So these viscoelastic properties are completely directly related to the actomyosin cytoskeleton. And one of the things that we should be asking then if we want to answer these questions is, how do these biomechanical characteristics change in healthy embryos that have a whole actomyosin cytoskeleton, and how do they change in these dysmorphic oocytes? So maybe biomechanics can tell us something about oocyte quality. And the last thing I want to show you, it's a single slide here. It's on the cytoskeleton and oocyte wound healing. It might be a little separate thing, but actually is related. And why do I say that? Because, another thing that we do every day in the lab when you do ICSI, I'm going to talk about ICSI here, is we inject the sperm into the oocyte. And when we use the needle to go inside, what we're making is a big hole here in the plasma membrane, right? You have all seen this hole, this funnel that forms. And then when you retract the needle and the sperm is inside already, you see that there's a hole being formed here. And, you don't think about it anymore, but you hope that it's going to close at some point and that the embryo is going to make it. So in reality, what happens here, it's a process of wound healing. But wounds occur in epithelia, in skin, in organs, not in single cells. Or do they occur in single cells? Well, there's one single paper, not in humans, but this is in Drosophila, the fruit fly, oocytes or early embryos that have shown this. So if you make a wound in this embryo, like here, this hole here, you have a plasma membrane here, and underneath you have the actomyosin cytoskeleton. The first thing you're gonna see is a hole that is gonna have an area, and this area is going to start expanding. So the hole is going to be opening after you make the wound, and then it's going to start the area is going to start decreasing. That means that the hole is going to start coming together. So it's going to start a phase of first expansion and then contraction until it forms a closure. And what happens during these phases is the accumulation of the actomyosin cytoskeleton, actin and myosin, which they are contractile structures that are going to bring together the plasma membrane to close this hole until it's completely healed, right? So the main thing is, do high quality oocytes that have a nice healthy cytoskeleton heal an ICSI wound better or faster than the ones that are dysmorphic or that have less quality? These are things that we don't know. And I think this is a nice thing that we can talk about to see how to approach these questions, sharing knowledge between a basic research lab and a clinical IVF lab in order to try to bring these technologies and see whether they can benefit the patients. So the talk today, it was about oocyte cytoskeleton and its association to IVF outcomes. So I show you that oocyte cytoskeleton is associated to oocyte morphology more than quality, morphology. That it is also associated, the actomyosin cytoskeleton, to biomechanical characteristics of oocytes, which are important for predicting blastocyst formation and live births. And also, the cytoskeleton, the actomyosin cytoskeleton, is associated to the wound healing process when we make ICSI, or at least we have evidence suggesting that this is the case. I show you this paper from Drosophila, but I think we could do it in mammalian oocytes and see whether we can identify which are the oocytes that have specific morphology, that have a specific structure, that heal faster, and do they give rise to better embryos. So with this, I would like to thank you and take any question that you may have. And thank you again for your attention and the invitation.
Thank you, Dr. Juan. It was an absolute honor to hear you speak today. You explained everything so beautifully and with so much of ease. I think no one could have done more justice to this topic than you.
Thank you very much.
And thank you so much.
Good evening, Nancy. I have one kind of observation. Can I say?
Yes, sir. Yes, ma'am. Yes, ma'am. Definitely.
Yeah. So, good evening, everyone. I'm Charulata from Hyderabad, and Joan, it is wonderful presentation, and really, it is every point is having some kind of message and understanding. Regarding your last slide about wound healing in the Drosophila, I think there are few papers in human oocytes that when we do the ICSI, there will be a channel dissolution. If the dissolution time is more than 1 minute, then the viability of embryo might be compromised. So there, and my observation also, I have done the parallel study, and particularly when you say that there is a negative impact, particularly where you observe the dark granular cytoplasm in the centrally located, in such case channel dissolution takes little longer.
Yeah.
And that will affect the viability of the embryos. And there was one paper in 2000 also, they has kind of concluded that such kind of embryos will be aneuploid, like particularly dark centrally cytoplasm, where you feel that it is a viscous, it is that aspiration of cytoplasm is a bit difficult.
Yeah. Yes, I think as you just mentioned now, if you see, we see this pattern, we can make the connections, right? And we can see the evidence. We just need to connect the dots. I think that would be like if we, the thing is to find the proper collaborations, right? But if you find a laboratory that does basic research and you address these questions, I think this is a very nice paper that can have a great impact on the field and can help us understand, okay, now we use artificial intelligence.
Correct.
And we know that exactly these features that the artificial intelligence is identifying, they relate to the cytoskeleton or they relate to organelles. So we trust what the artificial intelligence is telling us because we know already the physiology of the oocyte. Thank you very much. Yes,
Thank you.
It's a nice comment. Yes.
Yeah. I have a question, Dr. Johan. So this is with respect to wound healing. I think you mentioned in your slides with respect to wound healing in Drosophila, right? So was that an embryo level itself or an oocyte? Because you showed a very beautiful image where it occurs in 3 phases: the expansion, contraction, and closure. So where the cadre comes into play, actin and myosin. So was that in?
These are the early. Sorry, I interrupted you. Did you want to say something else?
I mean, I started with the Drosophila embryos before I jumped into human embryology. So I'm quite familiar with the embryos of Drosophila. So was this experiment done on the oocyte level or embryo level?
Okay. Yes, it is in the embryo level, but very early embryo in which there's still no cellularization. So these are very. This very early embryo is considered as a single cell.
Yeah.
Because it's right after fertilization. There's no, so Drosophila is different from humans. For those of you who don't know, once the oocyte starts being fertilized, chromosome starts dividing, but there's no division of cells. but
Right.
there's no cell membranes in each chromosome. So the chromosome starts just dividing, and they share this single plasma membrane of the whole embryo, early embryo. So
If I'm
this
not
can be
wrong,
a
if I'm
single
not
cell.
wrong, it is called as a syncytial trophoblast or something.
Exactly, a syncytial. Syncytial means that they are sharing the same cytoplasm. Although they are many nuclei, uh, um, um, the cell is the same. Yes. So this was in early embryos. Yes.
Because I remember my master days, like when I used to collect the Drosophila embryos, every day I used to
Yes.
collect in the morning, say 500 to 1,000 embryos from experiments.
Yes. Yes. No, it's a great model. We could try to do this in mouse embryos, for example, to look at the wound healing and how it closes and whether, adding drugs might benefit the healing process. And, or on dysmorphic mouse embryos and see whether, they close slower. There are many possibilities. I think this is an open field for understanding the physiology.
I think
Because
more or less the molecules or the players which are involved in this wound healing, more or less the same,
yeah,
because they're highly conserved throughout the evolution, starting
yeah,
from flight to mammals.
exactly. But one of the things that I wanted to emphasize is that also from the 1800s, an observation that is very important is that morphology, it doesn't always mean function. Or in more modern terms, morphology doesn't always mean physiology, right?
Yeah,
So even the embryos that look nice and healthy, they
doesn't mean
might
that they're
have,
healthy.
yeah, they might have some structures that or molecular things that they don't, yeah, that we're not able to see. So at least we're focusing right now on morphology, but as we learn the physiology, we're gonna be understanding a little bit better what these oocyte and embryo qualities really mean, right?
Yeah, and I think also with that, you mentioned about the biomechanics of the viability of the blastocyst, right? So,
Mhm.
I mean, those oocytes with blastocyst, right? Was that in mice model or was
Yeah,
it
in
in?
a combination. So they did it both in mice and humans.
Okay,
Yeah,
great.
and they actually linked it to some expression of some genes. It's just that the result was
Mm-hmm.
not so surprising. The genes that are less expressed in embryo in oocytes that give rise to non-viable embryos were related to cell division. Yeah,
So it was a biomechanics come proteomics come genomic study.
exactly. Yeah, and it's the only one. It's the only one. No one else has either taken it and say, okay, let's transform it into a tool. Maybe it's expensive because you need engineers and people, but like the tools to aspirate and do these measurements, they are in the IVF lab.
True.
We would just need to adapt them. I don't know. We need to explore that field, I think.
Yeah. Thank you so much.
Thank you.
Do we have any other question?
Can I ask a question, Nancy?
Yes, sir.
Thank you. Yuan, it was, as everyone mentioned, an excellent presentation.
Thank you.
I wish to go back again to very basic questions, not related particularly to this talk, but I just wanted to know how often do you see abnormal quality of oocytes in donor cycles, especially in your area?
Yeah. This is something that, at least in the Eugene lab, is not. The thing is, at the moment, the morphology of the oocytes has not been an important feature to consider in donation cycles, right?
Right.
Because everyone assumes that they are just good quality. Of course, they give rise to better, to more blastocysts, or at least to good quality blastocysts, and to an increased rate of pregnancies. But it's not something that, at least in the clinic where I work, that people check every day or they score it. Nowadays, they are using this artificial intelligence algorithm, future fertility, to score oocytes. and get a score to predict whether they're going to become a blastocyst. So now the use of this algorithm, like, to be honest, I don't know if future fertility works properly or not, but the advantage of using it is that now we're going to start recording these scores and the pictures of the oocytes that we use to predict whether they become a blastocyst or not. There are less. There are some papers that say that some of these abnormalities are related to the stimulation protocol. In Eugene, we use a very well-defined stimulation protocol, and specifically for donation cycles, it doesn't change much, so there's not much room of variation. But yes, I agree that in order to understand a little bit more of the physiology, we should be checking, extracting information on the morphology of the oocytes and its morphological grade. Yes.
So thank you for this elaborated answer. Why I asked this question particularly, because I wanted to connect my question to the previous talk about the dietary influence on the oocyte quality. And since you come from Mediterranean, so I wanted to highlight that particular thing, that it is not always the oocyte quality in the perspective of the morphology, but it is the physiology that matters.
Exactly.
And it is not that those who are following a particular type of diet, they always produce good quality oocytes. It is much more than what we just observe.
Yes. Definitely, yes.
Thank you very much.
If I can just add a line, Dr. Sanjay, please.
Yes.
Well, of course, I got an idea what you actually were taking the question towards. You were more concerned with the dietary considerations. But yes, apart from that, theoretically, we expect good quality oocytes from donors, but that is not what we get. And I believe it has more to do with the stimulation protocol that the banks are following to produce those oocytes. So I think stimulation protocol plays a major role as well, apart from the dietary considerations that you're talking about.
Yes.
So my concern is not just about the, dietary constraints. The thing is that, unfortunately, we have started living stagnant lives rather than practicing a good life. So we are sedentary now, and we are blaming our diet. So it is not just the diet, but the way we live.
Yeah.
Absolutely. Lifestyle, the lifestyle.
Actually, it's very important, this point, because mitochondria, they start dividing whenever they find the conditions that are important for them. When there's more energetic requirement from the cell, right? Sedentary lives do not require much of work, much of burning calories, energy. So even if we try to have a nice diet, what is going to make healthy mitochondria is constant movement, moving around, doing some manual work. We're all sitting at least I am sitting in the computer most of the whole day, 8 hours a day. It's definitely not healthy. And we have seen in the whole society how sperm concentrations have dropped in the last years. Oxidative stress levels, because it's not only sedentary but also cortisol levels that we're going through. We're not doing our best to keep healthy.
Absolutely. Thank you, Dr. Joan. Now, I think we should move towards the next session of our evening. Before moving towards the next session, we have something to share with you. And for that, I would like to call Dr. Ved Prakash sir to announce he is having a beautiful thing from iHera initiative for you all. Sir?
Thank you, Nancy. Actually, it is not so exciting, but yes, exciting because of iHera. are going to do something new, because you know that we are considering always blaming actually the female part. She is the male culprit, because of that infertility is there. But we are not considering about the male part, and of course the sperm. So we thought, why not to start with the sperm also? We have to think about sperm also deeply and understand what is going on there. For that, we are creating something new. For that, I invite Dr. Hetal Shukla, who is taking that initiative for that. And he is there, I think. Please, Dr. Nancy, call that Dr. Hetal Shukla to continue for that.
I think we already have sir with us. Sir, we can share the slides now.
Yes.
Thank you
So
so
good
much for
evening,
joining us.
everyone there. Hope my audio is clear
Yes, sir.
and the slide is visible.
No, sir, slide is not visible.
Let me share it just. I'll share. Now slide is visible, right?
Yes, sir.
Okay. So good evening, everyone, and a very warm welcome to the new initiative of iHera. Thank you very much, iHera, for bringing a new innovative initiative to our embryology community, to our clinical community of ART. And I have immense pleasure to announce a new initiative that is going to be known as Andronate. It is going to be a forum, an arm of iHera only, and this Andronet is making its philosophy of advancing andrology through science and education. So the ultimate aim is empowering male fertility professionally. So this Andronet, I have really great pleasure to invite you everyone with an idea. Why? Because clinical and laboratory excellence always dependent on quality control and standardization in andrology laboratory. Andrology labs require incorporation of advanced analysis, diagnostics, and many more things, improving ART success rates through enhanced andrology protocols, ethics, best practices in male reproductive medicine. This Andronet is having a mission statement, a pioneering scientific community dedicated to advancing andrology and reproductive. So this the main four focused area will be education and training, clinical excellence, research and innovation, awareness and advocacy, all these by bridging the gap between science, technology, and patient care. So Andronet will strive to be global leader in male reproductive health and andrology. Our aims are going to be community engagement and awareness by public awareness campaigns on male infertility, patient education and counseling programs, scientific publications and knowledge sharing, collaborations with IVF and ART centers for holistic fertility care. Innovations and research areas may be focused on, not limited to these, many more to come. But yes, to begin with, DNA fragmentation and male fertility studies, role of alpha glucosidase and other biomarkers, genomics, proteomics, anti-sperm antibody testing, immunological factors, integration of AI and machine learning in andrology labs. With future vision, expanding Andronet's global reach, fostering innovation in andrology, strengthening industry and academic partnerships, empowering the next generation andrologists. So this is our future vision. And with this, to make it more interactive, more successful, national, international, and global forum, this iHera initiative called as Andronet invites everyone with listed interests, not limited to these many more may be there. But yes, to begin with, I have listed some of that is routines and advanced semen analysis, DFI related studies, automated analysis and artificial intelligence, advanced semen preparation and sperm selection techniques, clinical andrology, management of erectile dysfunction, surgical retrieval of sperm tissues, lifestyle in relation to male infertility, genomics, proteomics, data analysis, manuscripting, ART banking, endocrinology, regenerative medicine in andrology, sperm biology, cryopreservation, sexually transmitted infections, and so many more to come. So these are just small effort to bring all interested andrology, embryology, clinicians, researchers, everyone can come together to this new initiative of iHera, which will be known as Andronet. Here is a link, email address, and a QR code which will help you to connect with Andronet with your interest, and with immense pleasure and with very good wishes. I am really pleased to convene this Andronet. Thank you very much, iHera. Thank you.
Thank you so much, sir. And a big congratulations to Team iHera and everyone connected. We are a community which is connected with every embryologist, junior, senior students. Everyone is welcome to join us at Andronet. And I think without taking much of the time, because everyone is waiting for the panel, let's move toward the next session of the evening, which is panel discussion. Sir, you need to stop share.
Let me stop share.
Thank you.
Thank you.
So just give me a second. So for the next session of the evening, we have a wonderful panel discussion with us. What are the clinical and the lab parameters that affect cytoskeleton of the gametes? And for that, we have wonderful set of panelists and moderators with us. And for moderating the session, we have Dr. Akash Agarwal and Dr. Sanket Dhumal. Dr. Akash Agarwal is a MBBS ASHRAE certified clinical embryologist. He was topper of 2019 batch. He is practicing male infertility and embryology since then. And right now he is a scientific director of Egde Fertility Hyderabad. And he's having many authorizations, and he co-authored many books. He's having many things under his name, not taking...
If I may interrupt, Nancy, I think you can make it fast. Don't read the introduction. Yeah.
And now we have with us Dr. Sanket Dhumal, sir, as a senior consultant embryologist, Apollo Fertility, Hyderabad. Sir, over to you. You can take over.
Yeah, thank you so much, Nancy. And I think we have a stellar panel. We don't need any introduction to anyone, but still, just to formally introduce for the newcomers, we have Dr. Vijay Kumar Chelur sir with us, who is MSc PhD and well-known embryologist, and we have Dr. Bindu Chimutta with us. Welcome, madam. And next we have Dr. Charulata Chatterjee, and also Dr. Sanjay Maheshwari, and Dr. Ashwini Kale, and we have Dr. Ananta Lakshmi, and we have Dr. Gaurav Mittal, and we have Karan. So all our dear friends and dear seniors. So it gives me absolute pleasure to moderate this panel. And I think my co-moderator is missing here. Anyways, we'll manage. And yeah, let me share my screen. Is my screen visible?
Go ahead, it's visible.
Okay, thank you. Thank you, sir. Well, after 3 lectures, which are completely dealt about cytoskeleton, let's have a panel discussion on the very topic. And yeah, I already introduced the panel. And just before jumping into lecture, I mean, the panel discussion, I'll just give you the glimpse of how it goes. The very topic what we are discussing, it is linked with 2 Nobel Prizes. I mean, a couple of more will be there. This is just to give the glimpse of the topic that we are going to discuss. In the 2001, it was awarded for Hartwell as well as Hunt and Nurse, where they discovered regulators of cell cycle, cyclins, and CDKs, which is again related to cytoskeleton and dynamics of cytoskeleton. And also in 2013, Rothman as well as Randy Shackman and as well as Sudov, the very common names that you cannot hear in the East cell biology or the basic biology books. They have discovered this mechanism regulating vesicular trafficking in cells, again directly related to this microfilaments and as well as microtubules with respect to cytoskeleton. So this is, I mean, there are a couple of more, maybe more directly or indirectly linking to the cytoskeleton. So this is the gravity of the topic that we're going to discuss. And I think those who heard the first lecture, I think I'm pretty sure Sindhuja has covered a lot of points. I have to remove a lot of points because it has been already dealt with by Sindhuja or the second speaker as well as third speaker. So I'll just give the glimpse for the newcomers, I mean, who just joined. So basically, cytoskeleton forms the structural framework where it provides the mechanical support and maintains the cell shape and also organizes the internal structures in both the gametes, that's oocyte and sperm. And also, it has a very key role in cell division as well as maturation, where it is crucial for the formation of the meiotic spindle formation and also the chromosome alignment and also the polar body extrusion, which has a direct impact on the fertilization potential and also the overall embryo quality. And as the earlier speaker said, it is not static, it is dynamic in nature, and it undergoes very constant reorganization during entire gametogenesis, say from maturation to fertilization, even during early embryogenesis, adapting to the very functional needs that for the each stage. And I just want to play a video how the microcellular catwalks look like. So this is the vesicle that is being carried by the kinesin proteins inside the cell, with the help of microtubules related to cytoskeleton. This is a motor protein. This is just imagine this entire factory that goes inside the cell. So so these micro catwalks of the cellular world are nothing but that's how the cellular logic works basically. And so far, there are 45 genes that are related to this been identified so far, which are involved in this molecular transport. And also, there are a lot of this choreography is basically guided by phosphorylation as well as G-coupled protein receptors and also the tempo of calcium ions. And this kinesin superfamily proteins, which is hub of family of proteins, forms a cargo from organelles to genetic blueprints along this microtubule highways. Having said that, let's jump to the first question. So this question is for all. Anyone can take up this question. It's basically a clinical question. So how do common clinical conditions such as PCOS and endometriosis or advanced maternal age affect the oocyte, especially with respect to cytoskeletal integrity?
Yeah, Sanket, can I take it? Ashwini here.
Yeah, Dr. Please.
So basically, these are 3 different conditions. One is PCOS, endometriosis, and advanced maternal age. And at the end of it, all of it results in a poor oocyte quality. So in PCOS, the main 3 factors that we see is typically hyperandrogenism, which affects your gene mechanism, motility, and the microtubule levels. Then there is hyperinsulinemia. So that is what is very important in PCOS. And finally, it affects the intrafollicular environment or the microenvironment, and which ultimately results in an immature oocyte. The maturity is delayed. And the third factor there is hyperexposure to high levels of estradiol, because since there is no maturation which is seen in this PCOS and there are too many antral follicles, because of PCOS. So ultimately, the oocyte maturity is affected there. Then comes endometriosis, where the basic physiology is the inflammatory pattern because it's a chronic inflammatory condition. And then you can see that it ultimately affects the mitochondria and it results in a bad quality of oocyte, wherein you see cytoplasmic abnormalities more. And then the third thing is advanced maternal age, which typically affects your microtubules or the meiotic division, where you see that the cytoskeletal division is hampered because ultimately the mitochondrial chain is hampered because there is less of CoQ in all of this, because it's an advancing maternal age. So that is why we see that typically in these patients, there are high chances of aneuploidy because the division that occurs, it is not equal, it is unequal.
Right.
And probably because of all of this, now a lot of NAD therapy is coming, which is even the precursor of CoQ, which ultimately helps in this electron transport chain. So that is what we were talking in the previous talk, probably of micronutrients. But now even further micronutrients have come, which actually help this electron transport chain.
Right. Wonderful, wonderful. So I think Dr. Ashwini gave the clinical aspects of it. I just want to hear the embryological part of it. How does it going to affect this?
If we can add, Sanket, to that, along with age, obesity as well, PCOS, endometriosis. So basically,
Also the
first of all,
combination of
can I talk, please?
Yeah, you're
Yeah.
already audible.
I mean, to begin with, this is a wonderful topic. We are trying to pick up an ocean in a nutshell. I mean, bring an ocean in a nutshell, because when we talk of the cytoskeleton structure, we are not just talking about the structure, the function. We are talking of the entire framework of what IVF is all about. I mean, we talk of oocytes in sperm and how maturation is affected, how fertilization is affected, and how cell division is affected. So the entire biochemistry, and as Dr. Sanjay Shukla mentioned, the physiology. So physiology and biochemistry, everything we are talking about in a nutshell. And as Ashwini very rightly pointed out, that with age, I mean, there is dissociation of centrosomal proteins, therefore disintegration of the meiotic spindle, and this results in the decreased maturation, decreased developmental potential. I mean, if we talk with respect to the embryologic parameters in the lab, so you have dissociation of centrosomal proteins, you have the meiotic spindle which is disintegrated, and therefore there are higher chances of aneuploidy with advanced age and decreased developmental potential. So you have maturity level also low and development also low. Similarly, with respect to I mean, obesity also, which has not been included, but then since we have included PCOS, it goes hand in hand. So the entire altogether. So the embryo mitochondrial metabolism is compromised in obesity because of hampering of lipid metabolism, right? So all this will affect the quality of the oocyte eventually, the maturity to begin with. And if in case they are mature, then the developmental potential further will be affected. So age, obesity, PCS, of course, there are so many other factors, hormonal imbalance and all, they all affect the oocyte maturation, lipid metabolism, as I said. And again here, elderly, obese women, meiotic spindle disrupted, and birth defects and miscarriage if pregnancy occurs. You are fortunate enough to go ahead. So there are higher chances of birth defects and miscarriages. And with respect to endometriosis, it is all because of the oxidative stress. So the oxidative stress again will cause spindle formation errors, which will lead to, I mean, there is high ROS, there is deterioration again in the mitochondrial activity, the telomere shortening, DNA damage, and so on. So it is a cascade mechanism that we are talking about. A single oxidative stress is not a small thing. It's a process by itself which will disrupt your spindle formation, and therefore it will lead to further developmental problems in your laboratory with respect to development from the oocyte to the blastocyst stage.
Absolutely, ma'am. And yeah, I mean, any other panelists would like to?
Just I want to add a point, ma'am. In PCOS, we have so many different phenotypes, phenotype A, B, C, and D. In that, I feel the patients with hyperandrogenism particularly will have a poor oocyte quality. Compared to hyperinsulinemia and hyperandrogenism, I feel it is more deteriorating to the oocyte.
With respect to the quality of the oocyte?
Yes.
Okay. Okay.
Sanket, I feel that whenever you denude an oocyte, you never feel that this is the exact picture copy of what you want. There is some or the other defect morphologically which you see and which you feel that probably this may not fertilize because the patient had PCOS, endometriosis, obesity, or advanced maternal age, recurrent IVF failures, recurrent pregnancy losses. So
And
I think
even
ultimately
the
it
very
is
stimulation protocol.
the stimulation protocol, true.
So that brings us to it's not just the nuclear maturation, the cytoplasmic man. So if you're talking of the cytoskeletal structure, we are
Combination of
basically
both.
also talking about not just the nuclear, but the cyto, because when we see endoplasmic reticulum, what is that? When we see a lot of granulation, there is no distribution of cytoplasmic organelles within the cytoplasm properly, and that is because of an error somewhere in the cytoskeleton. Because of various factors that
basically we talk about physiology, but we see only morphology.
Yes,
absolutely.
But then that morphology gives us an idea that what could be going wrong. I mean, a lot of granular cytoplasm, central granulation, presence of endoplasmic reticulum, all these are presence of vacuoles sometimes also, and the presence of those inclusions.
Cytoplasmic inclusions.
Sanket, in simple words, all these conditions will lead to physiological imbalances, physiological imbalances, which are observable in morphology too.
Yes.
Morphologically dependent physiological disturbances, all these will affect. And because a cell is made up of cytoskeletals, so in fact, we are able to see this very prominently in morphology, basically because the entire physiology is derailed. In all these conditions, the physiology is derailed. Now, how it affects, and everything, if we have to do it, we will have to do it in animal models, which we cannot say that it can be replicated in human beings. But we are seeing morphological implications of physiological imbalances, and that is, we know the reason that is brought about by this clinical condition.
Yes, yes.
Right,
and these are all your indications for the patient going IVF. So all the patients will show you these morphological changes.
In fact, most of the morphological features, you will not be able to observe it if you are doing it in IVF.
Yeah, because
it's only possible if you are injecting.
I think, sir.
Then at the oocyte stage, you can pick up certain subtle abnormalities, especially vacuoles, vesicles, inclusion bodies, centralized granularity. These are not visible in egg stage. You will be seeing it only if you have denuded.
So even our atlas for the quality of the oocyte speaks only about cytoplasmic features as well as extra cytoplasmic features. It doesn't include anything internal. I mean, it's all, as Ashwini rightly said, only morphology, nothing there.
Yes.
No physiology. Yeah. Having said that, I think thanks, Vijay sir, for summing up this first question. And going to second question, yeah, this is again a clinical question. Do we see any differences between?
I would take.
Yes.
Yeah, please, sir.
I would take it. I would. See, basically, what happens is, we progressed from agonist protocols to antagonist protocol. And most of our clinicians learn from medical representative. They don't most of our clinicians do not learn from the basic physiology part of it. They learn from the medical representatives and their supported material. And naturally, if I want to sell something, I will select only those papers which are favorable to me, not anti to me. Now, in agonist, we had 4 generations. First generation was buserelin, but buserelin had a problem. The problem was you need to start with a higher dose, then you need to reduce it to half the dose during the process and other things. And the downregulation plethora was actually tested biochemically rather than sonographically. You know, less than 40 picograms per ml of estradiol should be there to mark it as a properly downregulated, quiet ovaries on the sonography and biochemical values of estradiol being very less. You know, all these things were markers. Now, what has happened is people, after they moved on to antagonist, and then agonist came with the second generation, that was decapeptyl. Third generation was Zoladex. these two were so potent that they used to keep down regulation for up to 3 months. Zoladex keeps it for almost 90 days. So we started using them for endometriosis. Because that was a potent down regulant, pituitary down regulant. Then came the 4th generation leuprolide acetate. Now the story comes up here is the leuprolide acetate is very, very potent in down regulating, but at the same time is very, very cost effective, which does not provide any great money value to the company which is producing it. They came up with the antagonist. Antagonist initially was made exclusively for PCOS patients because in PCOS patients, agonist protocols were not very, very successful. We could not use HMC straightforward.
You mean to say it was marketed well?
It was marketed very well. It was marketed very well. Now, with antagonist also, we have so many variable protocols. There are some people who suggest that in the previous cycle, in the late luteal phase, you give 3 injections and then allow for this one. Then you go for a variable this one. You start at 12 millimeters, you start at 10 millimeters. But if you calculate it, the total cost in terms of injections, in terms of rupee value, what it generates, with respect to agonist and the level of control what it can provide, if you see it, it is almost 4 times expensive in certain patients. Okay. So rupee value is more, so it is being marketed more. But if you carefully look at the literature, do a slicing analysis with vis-a-vis, what I have noticed is antagonist after the introduction has shortened the stimulation cycle, has made more people to get into it, because of which we are not giving enough time. And moreover, you can say that people are not dependent on the biochemical pathways by any means. You know, they are not testing estradiol. They are depending only exclusively on sonography. Estrogen levels, estrogenization, luteinization, everything is based on the sonography itself. Nobody is getting an estradiol report, how it is oscillating between day 3 to day 5 to day 8 to day 10. No feedback mechanism is being not tested at all. They are just giving, okay, I am giving some relics. That's enough for it. So we will start this patient. If it returns back, I will start 1 day prior. That's the thing. And I have been noticing that, you know, initially when we used to do agonist cycles, we used to match it with the patient's own natural cycle.
Initially,
you know, you might say that it is 26 to 32 days. That's the cycle length, individualistic to certain patients. Fine. If a patient has got a 30-day cycle, I cannot plan her physiology is such that I will have to do the stimulation of 13 days only. She is programmed in her whole body is adjusted to that particular cycle. I can't say that I'm doing an 8-day stimulation there. So that has been taken off in antagonist, because of which after the introduction of antagonist, I am seeing higher incidences of centralized granularity. I am seeing very high incidence of smooth endoplasmic reticulum clusters being seen. I am seeing very high incidence of septate zona and a high perivitelline space.
That's PVS.
Sometimes debris in the perivitelline space too. So these abnormalities, which are straight away suggesting that there is certain maturity defect, cytoplasmic maturity defect, which is nothing but our cytoskeletal defects.
Vijay sir, probably these defects are also because of the fact that people are doing batching nowadays. And they straight away start with a fixed dose.
We have been doing batching from the day 1 of our lives.
No. Can I add something?
Even in the West,
it is also because using a higher dose of gonadotropins when it is actually not required. It is one dose
it was
for
not
all.
required even in
so
agonist
that is also.
See, it was not required in agonist also. All this started, the higher doses of gonadotropin started because of the egg sharing and egg donation business. It had nothing to do with this one.
The basic difference is that in the agonist cycle, there was comprehensive down regulation. There was a uniform cohort that we began with, and therefore the development of follicles was uniform.
And that is the reason many years back,
Bindu, because we did not want ovarian hyperstimulation syndrome,
Yes.
therefore this antac stimulation came up, and therefore they did not wait for comprehensive down regulation. Now there was a differential cohort, so they were all not of the same stage.
Absolutely, discordant cohort,
and therefore discordant follicles, and therefore now how does it affect with respect to what we are talking today? With respect to that, see, stimulation protocol, what it does is there is a distribution, differential distribution of mitochondria. So the pattern of mitochondrial distribution changes in agonist protocol because it was comprehensive down regulation, because there was a uniform cohort, because the dose therefore of stimulation gonadotrophins use was more or less fixed and it was maintained. So 10 days of stimulation, maybe, 150 IU was sufficient for a normal responder woman.
Absolutely.
And it went off well, and therefore the mitochondrial distribution, the maintenance of the cytoskeletal structure was maintained. Whereas in antagonist stimulation protocol, now, because there is no comprehensive down regulation, there are too many follicles of differential sizes, and as they come up, secondary tertiary follicles also keep coming up, and therefore there is mitochondrial hyperactivity. This in turn leads to all the maturation problems. So not just nuclear maturation, cytoplasmic maturation is because of this reason, is that all the follicles are differential, and secondary tertiary follicles coming up, therefore increasing the dose, no response. You start with 225, you start with 300, then to make it cheaper, you use HMG. So some people use up till 500, 600 IU of HMG.
Basically,
and
that's where I wanted
down regulation, PCOs were also maintained very well.
Correct.
So
with
the
the
dose
agonist
management
protocol, with the agonist protocol, now most of my consultants with whom I am working, they have returned back to agonist protocol.
Yes.
Nobody puts a thinking, till 2008, we didn't had any great problems with the stimulation. That's when the donor oocyte and donor stimulation started, and people started the direct starting dose itself is 300.
Yeah.
That was again an industry mediated, because the industry wanted to grow from 100 crore size to 1600 crore size. That was the target.
Yeah, absolutely. And therefore, when we say that the number and quality of oocytes is better in agonist cycle, it all boils down to this.
Yes,
quality
our
of
starting
oocytes
doses
is better.
used to be 150, and for some patients,
PCOS,
we used to start with 75
in
agonist protocol.
Yes.
But now you take anybody, even if the patient is 26 years age, the starting dose is 300, 375, 450. There is nothing below 300.
No, and what complicates this matter is because now we have a differential cohort of follicles, the timing of trigger. This further
you cannot just decide timing of trigger in antagonist.
Yeah. So it's
all that the people follow is 3 follicles at 18 millimeter, you induce the trigger. End of the story.
And then in that, the timing of trigger, the dose of
and
trigger,
the type of trigger and the dose of trigger.
Dual trigger, double trigger, all this we are unnecessarily complicating.
Unnecessary. It's all unnecessary. It's all unnecessary. It is iatrogenic.
Yes.
This entire thing is iatrogenic.
We should have some good clinicians also in this discussion.
So, no, they don't agree to this at all.
Excuse me. Good evening, everybody.
Hello. I will not agree to this, that it is industry driven. It is, we all learn from our experiences. That is why from agonist we shifted to antagonist, and now again we are going to agonist.
We have returned back to agonist. Most of my places are into
that is all from the experience and the kind of the oocytes that we are getting. It cannot be completely industry driven for sure.
Completely, of course, because we wanted to prevent OHSs. I mean, going towards an OHSS free clinic, that was the reason behind going for antagonist protocol.
So like in low ovarian reserve, initially they said that antagonist is the protocol of choice. Now they've again gone back saying that agonist is the protocol of choice. So it is again changed.
Simplest thing, madam, how did you recognize low ovarian reserve if the patient had come to you in the very first place?
Good evening, everybody. May I add something?
Yes, please.
Yes, sir.
Hello. We have discussed a lot why agonist started, why antagonist started, and where. Basically, I don't know single clinician who have ever started agonist or antagonist protocol to just look for this cytoskeleton abnormalities. They start with some different reasons. So it is no,
about
sir,
4 or 5
it is clear.
embryologists here.
It
Charu,
is very
Manju
clear.
madam, me, we
They
all
may be, sir.
communicate to our clinicians
Sir,
and we tell them that after
Let
the
us hear him out
came
what
up,
he's trying to say. Yeah,
Sir,
let us
sir,
hear him out.
you
Yeah.
may say we may talk about anything. These antagonist and agonist protocols are started depending upon their safety and requirement. But never, I have ever heard any clinician starting agonist or antagonist protocol to avoid any cytoskeletal abnormality.
No.
This is
It
a
was not
hard,
that. It is
this
the
is a
other
bitter
way
truth.
round.
You have
No.
to
After
let
seeing
me
this,
finish.
he found out.
No. Please,
Dear panelists,
let me
please
complete.
let's stop here.
I think
We're
Sanket,
going to
you
take
should
a lot
move
of
on
time
to the
into this.
next question
A
now.
very controversial topic of agonist
No,
versus
Dr.
antagonist.
Maheshwari
No,
is
no, no.
right. Dr.
Yeah.
Maheshwari is right. It is not the job of the clinician to study the cytoskeleton. What we
Correct.
are saying is that because of this protocol, we as embryologists see in the lab how the oocyte quality is affected.
we started communicating back to them
Correct.
saying
Sir,
that after you, what
actually,
kind of a protocol you are using, and I am seeing
sir,
this highly.
Right.
And
we have
when they started
protocol has
doing,
been
it's
already
our
started, and we
responsibility
are
also
getting
to
the oocyte.
communicate back.
Yes,
Sir, we may
you're
communicate
right,
back,
doctor.
but
I mean,
It's
yeah.
our duty to get back
All
at
said and
them.
done. All said and done. Yeah.
no, sir. Nobody listens to you.
That's what I said. No
That's
clinician
what is the
listens to
problem.
you.
That's what is the problem.
That's what
Nobody
is the
listens.
problem. I
You're
made
taking
it
the
at the
blame.
very first itself.
Sir, basically, we get the oocytes. So whether they are defected or not,
I
protocol
always
is
keep
not.
on saying that we are in the
The moment I was writing this question,
whatever I get, there will be certain reduction in that. There will definitely not be any addition into that. I have a defective product, I will be having a defective
I'm sorry.
embryo.
I'm sorry. I didn't like this sentence. Nobody listens to you. If you don't listen to the embryologist, I mean, we go hand in hand. How can we do that?
No, absolutely. But they don't change.
We'll stop here. I mean,
Yeah.
the moment I was writing this question, I thought a lot of noise will be there on this. That is evident, quite evident. Having said that, let's move on to the next aspect of it. I mean, let's come to the embryology part, hardcore embryology part of it. So, how critical is timing of oocyte denudation and also ICSI, the timings of ICSI with respect to preserving the cytoskeletal organization? Like, I mean, this is a very basic question, but it has very
yeah. So Sanket, thanks to taking the third question. So we have come over the kind of
Back to embryology.
never-ending debate on the agonist, antagonist, and so yeah. But they had that question was very interesting, and with we few of who are practicing from 90s as a good evidence to speak on. But let's move on. So this question is very basic, and timing of denudation is also like we all know that what time we should do the ICSI. But the thing is, denuding at the time of ICSI or before the ICSI, and what is the mechanical pressure and all that physical, it will have the effect on the oocyte. Like physically, it will be stressed, and few the diameter of denuding pipette 110, 120, 140, 170. It's so that comes into the picture. And ICSI already we have discussed in the third presentation that how it affects if we not good enough and very practiced embryology, we might damage the oocyte, or we might kind of means if it is polar body is not in the proper place, if it is not, then we might affect, and particularly when you are using the frozen thawed oocytes, so it might affect. So that is all things we have to be careful, and we have to follow the guidelines given by
So timing of oocyte denudation,
Istanbul. Yeah.
Means with respect to day of timing of trigger, do you calculate
Yeah.
with respect to timing of trigger? Does that affect?
So that's what we follow our 40 hours, kind of ICSI, and before
40
ICSI,
hours post trigger or after
post HCG.
post HCG.
Yeah.
Post
So
trigger.
39 to
trigger.
41 hours from post
Yeah.
HCG should be the
Post
injection
HCG.
time.
Yeah,
And
yeah.
our prior to
Absolutely.
that can be a denudation time.
Yeah. So prior to that, because we
There
allow
has to
some
be a
time with
gap
the
of at least
cumulus.
45 minutes to 60 minutes from
Absolutely.
denudation to ICSI.
So immediately, generally, some visiting embryologists immediately denude and keep it is we allow with the cumulus, so that kind of maturation happens. And as 40 minutes or 30, before the ICSI, we should denude. So it have some time to, stabilize the
So, ma'am, you
denuded
mean to
oocytes.
say,
If a person is planning,
you mean to
if
say,
a
ma'am,
person is planning
denude
for
the
a
oocytes,
oocyte freezing.
you mean to say denude the oocytes and start immediately ICSI? Don't have to.
No.
No,
You have
you
to
have,
keep for minimum
because
30,
you have
40
applied
minutes.
mechanical pressure, so you need some time for it to
For them to
relapse
regain
back.
the maturity.
Regain.
Regain
And
back,
it should, the
yeah.
denuded oocyte also should come to the, kind of means stabilize into the environment which after the denuding. So
I just want to
minimum
listen.
40 minutes.
Sanket, there is just a word here. If somebody is planning for an oocyte freezing for a future use, then the freezing should have been done at 38th hour from HCG.
Absolutely.
Because post-thawing, you will have to
warm it and allow it for 1 more or 2 more hours.
1 or 2 hours, that will coincide with 40th hour of HCG.
Correct.
So you will not be messing with the timelines.
But sir, we have a tendency of people allowing it for incubation for more period of time.
No, that is not
That
right.
is wrong.
If
Then
somebody
oocyte
has,
aging. That will be the oocyte aging again in the in vitro.
Yeah, because they wait till 40th hour to get it matured. Then, if it is matured now.
Afterwards, if you freeze it, then what happens is when you thaw it, you will be injecting it at 42nd or 43rd hour.
It's going to compromise the entire.
First, it is a cryo damage, and then it is a aging. 2 factors you will be introducing into
But,
it.
sir, but the point is, we're gonna think that we have put the biological clock into freeze. So
Yes, but after thawing, you are keeping it for 2 hours, 3 hours.
Exactly.
So the biological clock will start ticking.
Correct. So it's always recommended that you
38th hour from HCG should be the freezing time if you are planning it for a freezing for future use.
Correct. At least before 39th hour, you should freeze it off.
Yes, you should freeze it off 38 to 39 hours so that when you thaw it and leave it for about 2 hours for this one post thaw, it will still be within 39 to 41 hours of HCG.
As per the biological clock.
Okay,
as
so
per the biological clock.
Again, with respect to the cytoskeleton, what we are talking about, we all know that there is a lag in cytoplasmic maturation vis-a-vis the nuclear maturation in IVF.
Yes.
And therefore, see, when ovum pickup is done, on aspiration, supposedly oocytes are in the M2 stage. First polar body is already extruded. We have a M2 oocyte. We should be getting ideally. Right. So it is not about the nuclear maturation, but we are allowing it to remain for that much period of time after pickup so that the cytoplasmic organelles are reorganized and cytoplasmic maturation occurs. If we try to denude immediately after pickup, or say immediately after pickup is a misnomer because that depends upon what time.
What timing the pickup is
some
being
people do at 34 hours, some at 35 hours, some at 36 hours. But then all said and done, even if they do at 36 hours, and like Dr. Vijay mentioned, that some people visiting embryologists will immediately denude the oocytes. So in that process, we will have problem while injecting the oocyte because the cytoplasm is not mature. So cytoplasmic maturation, that means the reorganization of the structure, the placement of the spindle, all this is important when we say that we have to maintain the time.
Yeah, I mean, just coming back to that point, ma'am, just to extend the point, I think myself and Vijay sir had a discussion maybe more than 8 years ago for now. I was mentioning about the pressure that we're going to put when we're aspirating the follicle does have effect on cytoskeletal structure of the oocyte. Even though there is a proper cushioning with the camelus cells, it's going to affect it. So not only for maturation, also for relaxation or the reformation of whatever the structure, the original structure, we need to give time before ICSI. So, Dr. Vijay sir, please, could you explain that the pressure we're going to put at the time of aspiration?
Basically use a shorter diameter, small inner diameter of the injection needle. That's number one. There are ICSI needles which have got an inner diameter of 6 microns, 7 microns, all. Okay, but there are also ICSI needles which have got 4.5 microns also, 4.5 to 5 microns. Now, each manufacturer will have their own level of quality assurance programs. Plus or minus 1 micron is included into a matter. If somebody has kept it, and by chance, if you have taken a 7 micron ID, That can be 8 microns also and 6 microns also. If it is 6 microns, it is not a problem. If it is 8 microns, refer to Juan's talk. You know, the biomechanical aspects of it totally changes. The pressure and other thing totally changes. So whenever I teach people, I teach them only one thing, that you keep the cutoff as 50% equatorial plane in the injection side as your mark. The tip should not cross the 50% of the oocyte. Later on, you have to depend only on the injection, suction and injection pressure as gentle as possible. And the moment, and always take sperm tail first and inject head first. There is a reason to it. Tail is roughly around about 60 to 70 microns, whereas head is only 5 to 6 microns. And you should start withdrawing the injection needle the moment the head leaves out. With major portion of the tail is still inside the pipette, you start withdrawing, maintain the injection pressure, that's it. The injection is successfully done. You need not have to throw the sperm into the oocyte like a bullet fired from the pistol. That should not be the idea. So as gentle as possible so that you cause very least destruction. Even while denuding, people use, I have seen some people communicating to me saying that they use 125.
The ideal,
because the size of the oocyte is 100 to 110.
100 to 120, yeah.
Okay. So 125 they are using. Theoretically, that might be right. But the kind of pressures you will have to make it in order to suck with the corona cells surrounding it, you will have to exert more pressure, and that pressure will stretch the oocyte. So you are causing certain disturbances even during denudation. So the ideal oocyte denudation pipette would be 135 or 140, where you will be having least this one.
I think 140 is pretty much preferred, sir, because
135 to 140. Yeah, see, every company, 135 will not be 135, 140 will not be 140. Be sure of that. There is plus or minus certain variation. So my selection would be 135 to 140.
I mean, followed by 175 or 170?
Not needed at all. Not needed. Not needed. One pipette is sufficient for that.
They go with 175 or 170 first, and followed by 140 or 135. So
no, that is not needed. That is basically needed if you are directly putting it into hyaluronidase solution.
Okay. Yeah.
See, with practice in embryo, enzymology, if a person knows biochemistry well, they know that hyaluronidase acts on hyaluronan. So you take 1 ml of culture medium, you just add a drop of hyaluronidase, 80 IU per ml. That will practically cut down to almost 1 to 4 IU per ml. And it acts exclusively on hyaluronidase. And the moment the enzyme quantity is less, the timing has to be increased.
So we need
to leave it for around about 20 to 30 minutes. That's enough. The oocytes are beautifully denuded by itself with 1 or 2 layers of corona surrounding there. Once or twice you pull it into your pipette and your oocyte is denuded. You don't have to struggle much.
I've seen that, sir. I've seen that in UP.
Absolutely.
It is Vijay's practice. So here I'll comment one. I'll just tell one of my observation. So Vijay said very well that we just inject in the middle of the oocyte and then we take it out. So there is no cytoplasmic much of the disturbance. But a few cases where you observe that in a previous cycle there is compromised fertilization rate, and because of the oocyte activation, in few 2% of cases, if you go little 3/4 and aspirate the cytoplasm twice and a little manipulate, which is called a modified ICSI technique, I have observed that it improves the fertilization rate. So this is one of my observation for the cases has a previously little compromised fertilization rate, maybe because of the oocyte activation defects.
Yeah, and sometimes just for the simple reason that the oocyte you cannot break the membrane and you have to really
suck it hard, or you have to move it further.
That is mainly sometimes because of the viscous cytoplasm.
Yes. So
Bindu madam, there is one another trend which I am noticing. People fear to use spiked injection needles. They use blunt injection needles rather than spiked injection needles. That can also cause a problem for this. Spiked
Absolutely.
injection needles, just like our hypodermic needles, they pierce through very well and you get a ulemma properly. If you use a blunt needle, your suction and your suction pressure needs to be high in order to break that ulemma.
And also, sir, the wound will be more.
Cause more damage.
Wound will be more. Wound will be more, and it will not heal that faster either.
Absolutely.
That's why whenever somebody communicates to me with the problems of ICSI, I want them to send me a video. Send me what is the kind of the needle they have used and how was it done. That and all has to be analyzed. There is nothing called as out of the box answer for this. Spiked injection needles, I have seen most of the people are not using it because they go beyond the 50% mark with the piercing. That itself will create a wound again.
No, so you tell me what is your experience like with respect to, I've seen people who goes up to 9 o'clock position when they're
opposite side, yes.
Aspirate the opposite side. They even aspirate the
opposite side to double puncture, and because of the double puncture, the oocyte will spend more time in reverting back to its normalcy first.
The repair mechanism.
Repair mechanism, which will delay the entire events.
Absolutely.
True.
I mean, see, we are talking about this. We have to be careful about the whole cytoskeletal apparatus, the spindle, and we are talking about the calcium wave that is generated.
Yes, absolutely.
Yes.
Yeah, because, ma'am, as I said in the initial of the
gentler, purer, quicker way of doing it. End of the story.
Ma'am, the moment
gentle, sure, and
correct. Because I said in the earlier slides, in the very first slide, I said we won Nobel Prize in Physiology and Medicine because of this calcium oscillations, what we get to see with respect to cytoskeleton. If they are not happening, if you're introducing a lot of pressure and even the turmoil, I mean, that's going to affect the entire outcome. So you have to be as gentle as possible without disturbing the cytoskeleton as well as the other organelles which are cytoplasmic in nature.
I mean, oocyte activation is nothing but the calcium oscillations.
Yeah.
The orchestra of this calcium ions.
Yes, absolutely.
खाने के साथ खा लूंगा बहुत मीठी
संकेत
हमको भी भूख लगी है
जी सर
जस्ट अभी घर पहुंच जाओ संकेत वो है ना डॉक्टर संजीव एंड करण इज सिटिंग देयर.
No, sir, that questions are subjected to them because Karan is sitting quiet for some time.
Yeah, we'll keep quiet.
Probably our debate is happening too much. I think we all.
I'm actually enjoying to getting some knowledge from the seniors.
Gaurav, I had to leave for whatever reasons, and I just saw his message. Karan is sitting quiet for some time. Karan, this question is for you. Where are you? In India or UK? I don't know. Anyways.
No, I'm in London, brother. And I actually enjoying the debates from the seniors. So I was like quite sittingly, but I was like really thoroughly enjoying each perspective from the Vijay sir and the Bindu ma'am and everything. So it's fine.
Great. This question is for you. And yeah, could you please shed a light on this? How this lab factors, especially temperature fluctuations or pH variations or even the light exposure has an effect on the cytoskeleton?
I mean, definitely, if we are talking about in general of each gamete, either it's a oocyte or sperm or even the embryo, this correlation between the temperature, pH, and light exposure are in between, like correlate to each of these. So if we talk about the temperature, if it is below fold down 1 or 2 degrees, it's starting the depolymerization of the actin, and it will be affect to cytoskeleton of the oocyte. The same way, if we are talking about the pH and the CO2 levels, they both are intercorrelated. If it is the acidosis or alkalosis, it will be effect of the tubulins and the actin filaments, and it will be affect the total cytoskeleton of the oocyte. So ultimately, like they all are intercorrelated with each of them.
So what are the ideal recommendations for the temperature that you're gonna recommend for practitioners to practice ICSI or IVF during in vitro culture, and also the pH and also the light?
So temperature, we are ideally like talking about the 37, but then again we when we talk about the 37, then we need to think about our external thermometer as well, not just believing in what the temperature is showing from the hood or even the incubator. We have to use our secondary thermometer to check that is actually showing the 37 or not. And if we are talking about the pH, we would say like somewhere between 7.2 to 7.4, but the 7.4 is like the upper limit. So in our lab, we do the pH every quarterly, and we always believe that to
Quarterly,
do
you mean to
pH
say with
maximum of
respect
is
to batch of
7.3.
current quarterly, you mean to say with the batch of media going to receive quarterly, or is it a frequent measurement that you're going to do once in a while?
So frequent measurement once in a while.
Okay. With respect to every batch of media?
Every batch of media.
That's quite hectic, and I don't know how many people are doing it in India. I mean, that
But
usually
we have to follow
Even if
the
it
rules and
is
everything, so we are following that.
Right, right. Okay. And yeah, Sanjeev sir?
Yes,
What
sir.
comments are
I
on
also agree. I also agree with that. These are conditions like temperature, pH variation, they affect so much so that if there is any temperature fall, there is variation immediately during oocyte collection. Probably you will be getting more GB because it is the time from 34 to 38 hours when this GB breakdown occurs. If we have altered this to a condition, so cytoskeleton are varying, and we may not get GB breakdown and all that. So that GB rate may be increased, something like that. If these conditions differ, these are deteriorating immediately right from the oocyte collection onward. So these need to be very carefully handled. Earlier, we used to think GB is not the uh, because of lab. It is only a stimulation problem, but I don't agree to that extent. If we are not maintaining temperature, etc., pH during collection, probably we are not allowing GB to break down. So these all conditions are very, very important. How these work now, I came to cytoskeleton function and all that.
Yeah,
So these are very important things.
absolutely agree with you, sir, because temperature fluctuation is such a very subtle thing that going to happen. For example, if you take, I mean, forget about humans or mammals. If you take species like bees or ants and all, even 1.5 degrees Celsius gonna change the sex of the organism. So that important. Forget about cytoskeleton and the very distribution of the chromosomes happen with respect to temperature fluctuations, even the pH variations. So, I mean,
Moreover, duration of that variation should also is very, very important. If it is for a longer duration, say it is during the entire uh, oocyte screening, probably it will affect the result very badly.
True. Yeah, Bindu ma'am, please.
Yes, can I add something? See, when we are, especially with respect to temperature, so we are talking about heat stress. And again, with respect to the topic that we are discussing today, what does it cause? It may cause protein damage, stress to the endoplasmic reticulum, overall oxidative stress, mitochondrial damage, and DNA damage. And all this in turn may result in maturation failure or apoptosis. And now, as it was mentioned earlier, with respect to wound healing, again, the oocyte itself has this mechanism of heat shock. Therefore, there are heat shock proteins, right?
Yes.
So, if there is a minor change in temperature, especially at a lower temperature, the resumption will be, I mean, better if it is brought back to the 37 degrees centigrade again immediately. I mean, for example, there was a study done where
you
they
say about
exposed the
HSP
oocytes.
70, HSP
Yeah.
70 and all.
No. Higher temperature, of course, will cause complete
I
disruption.
mean to say heat shock protein 70 and the combination of the.
Yes. So now there was this very interesting study which was done where exposure of oocytes was done to room temperature, right? So
Yeah.
37, we are talking about 24 to 26 degrees centigrade at for 2 minutes, 10 minutes, 30 minutes, and whether it caused a disruption of the spindle. So it was found that just 2 minutes exposure, it did cause a disruption, but then it was resumed as soon as the
1 hour
temperature
later.
was
1 hour later, ma'am. If I
maintained. Yeah.
remember the paper.
But then 10 minutes and 30 minutes, it may cause irreversible damage to the spindle and the overall cytoskeleton. So reversal from a lower temperature is easier. Higher temperature, higher than 37 degrees centigrade, how much higher it goes, even for 2 minutes, it will cause irreversible damage. So higher temperature
I
is
mean,
more.
ma'am, not only the temperature, even the time duration.
Of course, that's what the time duration. So higher temperature, even for 2 minutes, may cause irreversible damage. Lower temperature lower means we are talking of room temperature. So even about 8 degrees difference, it can be recovered if it is brought back to 37 at within 2 minutes. But more than that, it will cause irreversible damage. I mean, that was one study done that I had come across.
No, I think we discussed the same study.
So same way, like I can tell you about the light. We have nicely covered, temperature and the pH. Light also is very important. I have seen few labs, they will just switch off all the lights during the oocyte aspiration. No, it is then few cases, no, little light is necessary, Bindu. Otherwise, I have seen other junior embryologists or mid-level embryologists, they'll increase the light of the microscope, which is more detrimental.
Yes.
Correct. So in that case, what you have to have little light in the room also, plus keep that light of the microscope fixed so that nobody can increase during the kind of handling of the gametes outside. So light will also otherwise, land up into induce the most oxidative stress, and ultimately, as per our today's topic, it will affect the cytoskeleton structure of the oocyte in some or other way, because it can increase the level of the oxidative stress.
Ma'am, to be
Not
frank,
just the intensity of light, even the green filters, yellow filters.
Absolutely.
We may use. So
yeah, I mean, basically, light is nothing but the photons of energy is nothing but temperature.
Correct.
Yes.
So it's one and the same in a different aspect. And having said that, yes.
When we started, we were of the opinion that we will be using red filters because we are covered in the blood. So whatever light that can reach there was only because of the red, this one range. So we used to keep red light. But red light is more hot.
And even the amount of photons, the wavelength of photons is more.
Yes. So we stopped it, and then some people came to me with xenon light source for this one and other thing. Now we have just given up everything. It is halogen, which is a yellow light source, and use a blue filter. That's the end of it. Mono wavelength with a disturbed photon. That's sufficient.
What about LED? Any of the panelists, halogen versus LED?
At least I am not using it. At least I am not using it.
Okay.
I mean, as a source, you are saying?
Yeah, ma'am.
You
So
mean in the lab? No. But in the lab, installation is of LED lights. But then, as I said, it is only...
It's very minimal light. Only one light in a room at a distant corner from my stereoscope microscope would be on.
Yes.
Yes. That is neutral for all the procedures.
The best would be if you have natural light, if you have a glass through which you are getting natural
diffuse light. Diffuse light.
Yeah.
Just put a
Because
film onto that.
sometimes
It's
what
a
happens
diffuse
if
light.
it is totally dark, the person who is observing the oocytes under microscope will not be affected. But if you ask some person that get the dish from the incubator, they will be searching blindly. So I always keep one light distance on.
Especially when you're handling number of cases.
Yes.
Absolutely.
Yeah. Well, so the nutshell is that yes, temperature, the pH variations, and even the light exposure has effect in cytoskeleton. So we have to be in the limitations for whatever the recommendations has been there for the guidelines are there for temperature, pH as well as light. Having said that, and yeah, this is like a very tricky question, but I mean, no one has experimented on this, or I don't know. I just want to listen from the panel. So during the pipetting, the pressure does really matter, and even the exposure to enzymes like hyaluronidase at the time of denudation. Do we induce any cytoskeletal damage?
We always. Pressure, always. That's what I said. Use a right kind of a pipette and be as gentle as possible. And when it comes to enzymes, study biochemistry first, the enzymology first. Enzymes are specific. It is lock and key mechanism. They do not act onto other things unnecessarily. So don't go by those veracious claims that if you keep it in 80 IU per ml, this one for a longer time than 1 minute, you will lose the oocyte, the zona will harden. That and all is not going to happen. And minimize the amount of enzyme what you are using.
Yeah.
There is no need for you to go for an 80 IU straight forward. It is not needed.
Sir,
You need not have to
basically,
rush against time
no,
and say 1 minute and keep it there, wash it
correct.
8
Sir,
times, 10 times. So 8 times,
because,
10 times washing is automatically you are increasing the pipetting pressure.
sir, I've seen people who uses the ready-to-use media, even the hyaluronidase enzyme, they dilute with HEPAs or MOPs. So
Yes.
they take 0.5 of ready-to-use.
No. That is actually only Vitrolife provides you that. That is a 10 times concentrated.
No,
It is
no,
10x
sir.
concentrated.
No, sir. My point is different. I mean, we have to prepare for Vitrolife. Other companies, they do provide ready-to-use media,
Yeah,
right?
80 IU per ml ready to
But
use.
still, people, in spite of that, they dilute it with heaps or mops.
No,
So
I usually dilute it.
Yeah.
correct.
I take 1 ml of culture medium, put all my oocytes into that, and I just put a drop of it. A drop of it of less than 20
That's a
microliters,
different
that comes to
protocol.
practically 4 IU per ml.
Correct. If you calculate, if you know the enzyme kinetics, you can do that.
Yes. 5
So,
ml, 4 to 5 micro IU per ml is sufficient. And the moment the enzyme concentration goes down, you have to increase the time.
That's all. Instead of 30 seconds, 1 minute, you have to increase it for as per the dilution.
Up to 30 minutes I have kept.
I mean, 4 into 10 is 40. Yeah, you can keep for 20
Up to
plus
30 minutes I have kept and nothing has happened.
I've seen that, sir.
It's
Anyways.
equally safe.
And I mean, Bindu
Of course, I don't keep for all the patients for 30 minutes. That's for sure. It's usually 10 to 15 minutes. The job is done.
Correct. Bindu ma'am, Ashwini or Charu ma'am.
Sir, I want to add a point here.
Please.
When we are talking about pressure, we want to take care about the oocyte aspiration pressure too.
That's what I said.
Absolutely. There at
during
the bone pickup, the theater also. Yes, absolutely.
So you also have to understand that if the oocyte is not mature and it's not going to denude, you have to stop applying that pressure and accept it that probably that cumulus is there to stay. Because at time it happens that few are not denuded completely and you start increasing the pressure or you start increasing the time to denude it.
Especially
even I believe complete denuding is also not necessary. If you know little cumulus is there is another thought of school that if you keep it, you can see the polar body. It is fine.
And oocyte cytoplasm properly, that much denudation is
absolutely
sufficient.
Generally, the mature oocytes denude so easily.
Doesn't matter much.
Yeah.
As
so
long
once
as
you know
see the polar body, that's fine.
Yeah, once if you keep in a exceedis, you can rotate and you can see the polar body. So little
the action will be there will be some remnant action and that will help you to anyway get rid of.
Absolutely.
So the point is, how do we optimize these protocols with respect to pipetting pressure as well as the exposure to enzymes?
But before that, I think Anant Lakshmi mentioned a very good point. We had been talking about the temperature and everything in the lab during the time of pickup itself, and from the transfer from the OT to the lab. The maintenance of that temperature is absolutely necessary.
If you remember, a company came up with a patent saying that, I mean, the box that carries from OT to the tubes that from OT to lab, we have to maintain 37 degrees Celsius. I mean, they have taken a patent, I think. So, have any of you used those, and what are the differences there to get to see?
We came up with mobile nest in Shivani.
Correct, sir. Yeah. I mean, what is your experience with that, sir? Like, I mean,
it doesn't cause much of a difference even if you are transferring it by hand mode also. Provided if you have kept your tubes in preheated condition at the OT and you collect it into that. Don't hold it in air and try to collect and see what it is there.
And other thing is, distance from OT to IVF lab. I have seen
as minimal as
possible.
Absolutely. The distance needs to be as minimal as possible. And the moment you get it across, put it into the warm tube.
Yeah.
You might have taken 3 to 4 ml into a dish for scanning. So the remaining should be capped and kept in the warmer.
So unfortunately, that doesn't happen most of the labs. What they do is they do air collection. The tube will be in the hand of the nurse or a technician who is assisting the clinician. So I mean, these are the factors that has to be considered, because all the time the blame comes to the lab, embryology lab.
But I think everyone uses warmer blocks.
Warmer, even in the OT theater,
Even
they
in
will
the
be
OT,
warmer.
they have a
At
warmer.
times,
No,
madam,
Bindu
when there are
madam,
new
Bindu
people,
madam,
they take out the tube to
there
see whether it is
certain
filled or not.
places. There are certain
Stays in the
places.
block, and the
Blocks
block
will be
is
there, ma'am.
transferred.
Blocks will be there, ma'am.
There
What
are
they
certain
do is
places where, in order
they
to have this one, the warmer is not switched on. Just 10 or 15 minutes prior to the process, the entire system gets on. It would not have been equilibrated either properly. And they take out fresh tubes from the packet and put it into the warmer just then.
Those
It's
are
not
the
pre-warmed.
yeah, correct.
It's not
Correct.
pre-warmed.
They're not pre-warmed. Yeah. and I've seen people taking out the tubes from the warmer, and they do not, I mean, they're gonna do have
And then they just hold it like this just to see
No,
there is
but
no
there are,
overflow.
yeah, there are such other QC oriented theater also that I have seen, even dry incubator in the OT, where the pickup, before the pickup, they'll keep, even pickup needle and
media
cleaning
and the
distilled
tubes,
water,
the tubes in
saline,
there.
and everything, tubes in the warmer. So as per the calculation of the
I have
cases.
also witnessed one place where they had called me for troubleshooting. You know what they were doing? The clinic, the distance between the lab and the IVF OT was quite long. So what they used to do was, they used to finish the entire pickup and put it into these blocks and other things. Then the whole tray used to be transferred into the laboratory.
Most of the times you end up in
First follicle to last follicle. Imagine if a lady has around about 20 follicles, depending upon any surgical troubles they might face for accessing the follicle and other things and everything, it could be as high as 20 minutes.
Yeah, more than that, sir, times. And I mean, having said
I
that,
think this happens more when people do not flush the follicles quick. I mean, just aspirate the follicles, collect not even in the tube, in a beaker, and transfer that beaker to the lab.
One or two follicles per tube should be your optimization of the OPO this one, and immediately transfer that tube to the lab as quickly as possible.
Yeah, which is ideally recommended.
Yes.
Bindu ma'am, I think you were mentioning about the conical flask, right? People does collect at least 30 micro
Beaker, in a beaker, not conical flask.
Conical fluid for the skin, right? Yeah. Anyways, so I think it is very important when you're pipetting the oocytes or the other gametes, including the sperm, and also exposure to the enzymes, because it does have effect on cytoskeletal damage, because the more the time the enzymes or even slightly increase in the pressure, going to affect the very cytoskeleton. Having said that, yeah, this is like, I mean, this is like game changer for us, the cryopreservation, especially vitrification. But what are the effects of this cryoprotectants or the very vitrification process with respect to integrity of cytoskeleton, both in gametes as well as embryos?
So
Damages,
first thing
definitely
we have discussed earlier that oocyte, in case of oocyte freezing, after the trigger 38 hours, so that after warming we can have some time for ICSI. So that is related to oocyte, plus oocyte is also very sensitive to cryoprotectant, and so whatever like commercially available, we have to, any embryologist should read the literature and follow the protocol very step by step, so that exposure to the cryoprotectant will be not much. So that is related to oocyte, and many people have seen that freeze 6-7 oocyte in one straw. So I said that do not do that. So that's what
I think following any cryopreservation protocol by the company to the T is the key to it.
Yeah.
Absolutely.
Not altering it or changing it as per
Absolutely.
our wish.
Absolutely.
Yeah.
And mostly when we say that after thawing, we are waiting for 1 or 2 hours before doing ICSI is not just the reorganization, but also the spindle position.
Yes.
I mean, that's why people used Polscope, those who could afford,
Poloscope,
used
yeah.
Polscope to detect the position of the spindle, not just the
Spindle.
polar body. So the polar body may be there, but the spindle position may be disturbed because of
No, during pipetting in the zona, actually the polar body moves.
yes.
Polar body is free, that moves. So
Yes.
the original principle that the spindle will be quite next
Below
to the
the-
polar body might not hold good there.
Yes, yes. So
That is why there is a
I also wanted to know from the panelists, like, no, in nature, it doesn't usually happen, right? The shrinking of the gametes or the embryos at the time of vitrification, even the rewarming. So we are going to see the series of changes that the entire cell gonna shrunk, and even the expansion does happen. I mean, how does this gonna affect the cytoskeleton?
See, it will definitely affect. That's why not all oocytes which we freeze will thaw back
Survive.
to proper potential. It will definitely, but the thing is that morphologically we cannot observe such great difficulties in terms of that. So we need to have an investigative model to check through that. We don't have a direct evidence.
Yeah, because that's very surprising, because
I did the Polscope experiments to myself with only birefringence pattern with the cryopreserved oocytes.
Yeah,
They do
that is,
gain
yeah.
back to normalcy within 3 hours.
60 to 90 minutes.
Yeah.
Huh?
60 to 90 minutes.
No, 60 to 180 minutes.
Okay.
60 to 180 minutes. Yes, absolutely.
That means if you're freezing at 38 or 39th hour, your ICSI goes up to 42, 42nd
41 hours. 41 hours.
41 to 42.
Now, 42 is very rare, but 41 hours you can start the ICSI.
like Sir said, at 38th hour you freeze, then
You
if
have to
wait
freeze.
maximum 3 hours, that means 41 hours. 39, 40, 41.
41, yeah.
And to
But
add a
one
point,
thing sorry.
sorry, ma'am.
Sorry, please.
No,
Ananta,
we are
please go
talking much about the oocytes, ma'am. We want to take care of the embryos, cleavage stage embryo and the blastocyst. Many of the centers and there is a protocol to wait for after thawing the blastocyst for minimum of half an hour to 2 hours.
Up to 4 hours.
Confirm the re-expansion. That is also for the cytoskeleton recovery, I think.
I mean, not only that, re-expansion for survival as well as re-expansion.
To confirm the survival and also to check for it.
Basically, there are two types of survival. One is structural survival and the other one is the functional survival. Now, when we were doing slow freezing, we never used to freeze oocytes. We never used to freeze blastocysts.
Right.
We were only doing it in a cleavage stage. That was the one when we were doing slow freezing. And our assessment used to be if 50% of the blastocysts survived. I am not talking about this one, blastomeres. If I am freezing an 8-cell embryo, if 4 of the blastomeres have survived, then we used to call it as a survived embryo. But they might, what we used to do was, I still keep practicing the same. You freeze it on a day 3, thaw the embryos, allow them to go to blastocyst. If the functionality has revived, then it will definitely move forward.
Yes.
But in case of a blastocyst, the timing is shortened. Whether you do artificial collapse or this one, whatever you do prior to your vitrification process, invariably it collapses and it would have re-expanded. When you warm it also, it collapses first and then allow it to re-expand so that it can at least show that it is survived.
So yeah, because sir, the collapse and as well as the re-expansion is quite natural process for the blastocyst.
Yes.
Just to push for the hatching process. I mean, that's what we observe.
It is punching from in the punching bag from inside. That's it.
Yeah, I mean, that's what we hypothesize as of now. We don't know the exact reason why the blastocyst does collapse before the hatching.
And that's because of the same. Because when it collapses and when it re-expands, the timing can be something around about 4 to 16 hours for the total cycle to occur. By the same time, it would have added more cells because of the cell cycle. So the increased mass will start thinning out the external zona. And the point of entry of the sperm, zona repairs, but it will not repair in such a manner that the entire point where either your ICSI needle has gone or your sperm has gone, that part will not be repaired completely.
Sir, I
So
think
that's a weak spot. That's a weak spot.
So I
This
think
increased cell with collapse and re-expand with addition of the cells in between will start expanding that particular breach in
Start
zona
protruding.
to a greater extent, and that's where it just escapes.
I mean, sir,
So,
yeah, ma'am.
Yes, Sanket, are you talking about a normal expansion and contraction, or are you talking only with respect to
No,
frozen
normal.
thought?
In normal, ma'am.
So normal also, that is called breathing. Like that process is called breathing, where the blastocyst contracts and expands, and at that time you start seeing the appearance of zona breaker cells. So see, at the point of hatching, you see those small bubble-like structures coming up, right? They are called as the zona breaker cells. So that is the point of hatching. Just as like in a marathon, before if you have to run ahead, you go back a bit. So that similar is the process before causing a breach in the zona. The blastocyst, it contracts, expands, and that process is called breathing. During that time, there is the appearance of zona breaker cells. They are nothing but, there will be enzymes which will help in breaching the zona, and then hatching will begin.
But ma'am, we all noticed that it doesn't happen usually in all the blastocysts, only in some. Most of the blastocysts, when the evidence of time lapse also, they keep without the collapse, they start hatching. So it's a very unique observation that anyone could have done this as of now, because blastocyst.
See, initially the method we used to follow for assessment of a blastocyst and other things was counting the number of cells. If you happen to see WHO manual, there was also a method described to count the number of inner cell mass cells, the trophectoderm cells, and other things. For a better implanted blastocyst, the number of cells used to be 128 to 256.
220, 250, they say,
128 to 256, because it's doubling all the time. So whenever the cell mass, if you consider this Gardner's grading system, 2AA, 3AA, all these things, they will be, if you open that blastocyst, spread it on this one and do a differential staining of inner cell mass and this one, you will notice 64 to 90 cells only in those. So there is no mass in that to break open.
You mean to say in ICM, sir?
Not in ICM, the entire blastocyst I am talking about.
Including the trophectoderm.
Yes. There is a method to stain that. Count inner cell mass as well as trophectoderm cells. If we artificially open the zona, spread this one, but of course that embryo cannot be used.
We can do simple Giemsa banding.
Yeah, Giemsa banding is only nuclear wise. There is a differential staining method itself. So we used to do that, but the trouble with that is you will not be able to use that blastocyst. We used to use this on our leftover things.
Got it.
We used to use it on leftover things, and we used to count it. The poor grade blastocyst used to have very less number of cells. Sometimes it will be 64 cells only.
Okay, that's quite interesting.
Yeah, that's why the measurement came up. Measure the size at the equatorial plane of the blastocyst. What is this one? Say, for example, if somebody is having almost everyone has this videography connected to this one. So if you have got a 15-inch monitor and if you keep it at 20x objective without any intermittent change, like 1.6 times or anything optical boost, just keep it at in your eyepiece you will be seeing 200x, and if you see it with the camera there, if the zona of oocyte is not fitting into this one in the frame, that means that's a very good expansion, and that will usually have, I am not saying exactly, that might usually have roughly around about 200 to 225 cells.
Okay.
They are all old school people, Sanket.
No, that's absolutely great
No,
because
we have
We measure
graduated
the
from the old
diameter
school to the
also.
new
See,
school. We have
I mean,
adopted
where
to
we
that
cannot
also.
measure the number of cells, we can measure the diameter
correct.
and the inner
Diameter
cell
and get
mass.
across.
Yeah. So that
I
is it.
mean, it's. I mean, I'm little
Short of
privileged
time.
to have between the old school and new school, so I quite understand with the old school techniques as well as new school techniques. Having said that, moving on. Yeah, I mean, this is again a very tricky question. We don't know how it's gonna work, but we do incubate the embryos for a lot of time, the gametes from after ICSI or before ICSI also. So, how are these gas levels, CO2 or O2? Again, it comes boiled down to the very topic of pH. So, how does this gonna affect the uniformity of microtubules?
So again, as you said, it related to pH. So suppose more or less, I think everyone is using triple gas only. But even I said even CO2 big box incubator is also good enough to give the blastocyst also. But again, CO2 percentage of the related to the media which you are using and what are the pH suggested by that. According to that pH variation we discussed earlier, that how
And
it
the place
affects
where
the
you
cytoskeleton. Yeah, so sometimes even I have seen that incubator is kept under the HVAC system, so the temperature counts. So the position of the incubator in the lab is also very important, and definitely O2 and CO2 level is also very important into the keeping the cytoskeleton structure in the mind,
And see,
because it
when you're talking
reflects the
of
integrity of the microtubule basically.
Okay.
see,
So,
if denuded oocytes are very susceptible to any pH changes, temperature as well as pH changes.
correct.
When we say that after denudation we are keeping for a while before we do ICSI, you have to remember not to exceed the time, because denude as long as the cumulus is there, they are the internal pH and the external pH.
Is
Right?
maintained.
But then once you have denuded, the oocytes become very susceptible to any changes. So therefore, I mean, I personally do not prefer keeping it for even half an hour. After I have denuded, before I make any other thing, just 10 or 15 minutes is too much for me. I do ICSI after about 10 to 15 minutes after denudation. I mean, not half an hour or so, because somehow I feel that the denuded oocytes are very susceptible to temperature and pH changes.
Yeah, because I've seen people taking the oocytes directly into ICSI dish when, I mean, they might be using Hepicel mops for the as a buffer. But what they say is the time taken for the sperm selection. For example, you're taking 10 oocytes in a dish immediately after denudation. So they need at least 5 to 8 minutes to select a good spermatozoa, at least 5 minutes. So they say that that is more than enough because they're seeing better results. I mean, good results when compared to those who incubate the oocytes post denudation. So, I mean, this is what I heard. I mean, any comments from the panelist? So, ideally, panelist does recommend that post denudation, there has to be certain time for incubation before you take them into a oocyte, I mean, ICSI dish.
At least I propose that.
I don't keep for a longer time, but then yes, because I don't shift it directly after denudation into an ICSI dish with containing the buffer, but I put it again in the culture media. Maybe that's a way of washing again after I have denuded the and then place it. Yeah, and then place it. I check my ICSI dish, I check my suctions and everything, and then take up. So that is about it, but never half an hour for sure after denudation.
Okay. And this for all, I mean, are there any biomarkers or molecular indicators that helps us to assess the cytoskeleton? I mean, the health of the cytoskeleton in
As
nature.
of now, we do not
If
have
they
any
would have been there, probably we would have 100%
As
results.
of now, there are no biomarkers or molecular indicators which can be assessed during our functioning. There will be definitely, but if we use that, then the oocyte renders useless.
I mean, except the poloscope or something, which is indirect.
Say, poloscope can just give you an indication on spindle to some extent if you have maintained temperature properly. Birefringence of the zona is most probably visible, but that's not an indicator truly of a cytoskeletal health.
Right.
It's only one part of a tubule. The
Right.
other microtubules, it is not possible.
Yeah, because sir, having said that, it's very dynamic in nature, as we discussed from the first lecture.
Even spindle itself is dynamic in nature. Even if you maintain it at 37 degrees centigrade, we created a small globos, this one, and we kept a thermistor on the top and we started maintaining at 37. The spindle is also very dynamic. It doesn't always remain in a fibrous this one like the same throughout. In a process of 12 hours of observation, it went down and came back at least 6 or 7 times.
Yeah, probably, sir. Yeah.
So you cannot judge based on that same.
See, that is a very tricky question because assessing the cytoskeletal health, see, even if it is indirect, but the morphologically, the appearance of the cytoplasm that we see, that could be the only indicator. Because as Dr. Vijay Kumar said, once we start assessing, that means we are rendering the oocyte useless. There is no way how can there be an external parameter to judge the cytoskeleton? I mean, actin, microtubules and everything.
Yeah, I understand.
There are many
Biomarker
biomarkers, madam, but then they're all in the experimental basis.
No. Many biomarkers are not for cytoskeleton. There can be ROS. For ROS, there are. But then not that again is indirect.
See, the moment I put it into that
cytoskeleton you
biomarker
cannot
containing medium, then I am rendering that oocyte useless.
Correct. So cytoskeleton directly there cannot be.
No,
There can
sir.
always be indirect
indirect measures.
No. Let me
There
come
are
to this
marks.
point. No, let me come to this point. The question was made with a purpose saying that biomarkers not only with pertaining to the staining techniques, it could be you can aspirate, for example, aspirate a picolaters, whatever it is, from the oocyte and look into the proteins which are active. For example, say actin or even the proteins which
can
are.
measure the
I
caspases
completely
also.
understand the portion from where we are aspirating. If that is not having a proper distribution matrix in the oocyte, and the where I have taken out, it is the same thing what we have it with the PGT also. You take a trophectoderm biopsy at a particular spot where you have syncytial or trophectoderm. So you will have multiple nuclei. So it's the same trouble.
Mitochondrial copy number can be deduced.
Assess
mitochondrial copy
again,
number.
you are compromising with the oocyte there. You are reducing its potential.
No, sir.
So that's why I said the only biomarker, as far as I understand, is morphological observational biomarker. There we can pick up only 2 of the aspects: smooth endoplasmic clusters and centralized granularity.
So morphological in nature.
Morphological in nature. That's a cytoskeletal defect only. That can be picked up only if the oocyte shows these 2. That's it. Otherwise, you cannot be conclusively taking out.
Yeah, right, sir. And with
maybe AI can come up with something, we never know.
Maybe, ma'am, we don't know. As you rightly mentioned, even the mitochondria.
Mitochondrial copy number.
Yeah, even it's a bottleneck theory that we don't know that each oocyte will differ from 1 lakh to 10 lakh based on the oocyte. So, Dr. Yuvan said in this lecture, sir, that whenever there's a need for the cell, the mitochondria does duplicate on its own because they're autonomous bodies. For example, it takes hardly, say, 15 to 20 minutes to duplicate in thousands. So, I mean,
stress again makes it, hyper.
Oxidative stress does make an impact on it. So, I mean to say, having said that, it's again a long discussion. Let's not go into that. And impact of oxidative stress, just we're discussing about oxidative stress and ROS. How does this going to affect on the cytoskeleton in sperms as well as oocytes? Because the moment a sperm, it's highly dependent on the microtubules, which is part of cytoskeleton. If there is no good flagellar movement, then the sperm is immotile and it becomes difficult for an embryologist to choose whether the sperm is viable or non-viable. We have other factors for that, but still, integral part of it, oocytes as well as sperms, how does it going to affect with respect to OS?
See, for oocyte, I already mentioned, I think in the first question itself, where you talked about endometriosis. So whether it is tuberculosis, endometriosis, or a longer exposure time while handling the gametes, all that leads to oxidative stress. And as I said, it can affect in different ways, mainly the spindle formation error, the telomere shortening, the DNA damage, and the deterioration in the mitochondria, and of course, therefore, protein degradation, and therefore it will all lead to क्या बोलते हैं?
Cellular damage.
I mean cellular damage.
So what about lab induced or
Yes.
lab created oxidative stress?
Lab induced as well
Or
as
the
because of
sperm?
etiological. I mean the tuberculosis and endometriosis. If patients they already come with oxidative stress. I mean the oocytes are under oxidative stress, and therefore the quality of oocytes that we see is compromised in these patients.
Right.
And
Sanketa,
sperm
you
is
meant the semen preparation methods when you said that lab induced. So yes, probably there is a theory that it may induce oxidative damage.
No, oxidative damage is iatrogenic in terms of sperm preparation, because people go with funny modules saying that 2000 RPM, 2500 RPM and all. So don't measure your centrifugation with the 2000 RPM, 1500 RPM in RPM methods. You can calculate your G-force of your
radius of your
and your maximum G-force which you can apply to your this one is
300
250 G for 10 minutes.
300
That's
to
it.
400 G.
Yeah, would be optimal.
So we have done this where before and after centrifugation, right? The not the ROS as such, but then we measured the DNA fragmentation index post and pre centrifugation.
Yes.
Centrifugal force itself may cause oxidative damage. So, like Dr. Vijay said, that it is not about the speed, more the friction, and more oxidative stress on the sperm as well. So you have to come at a compromise.
Also, the
between
timing
speed
of
and
collection, preparing the sample, all this also introduces stress unknowingly.
As long as seminal plasma is there, you are protected. The moment you remove the seminal plasma, you induce stress.
I think that's the whole USP for microfluidics and stuff like that.
No,
I mean,
microfluidics, I don't even trust it. Forget
no,
it.
as Dr.
I don't even
Ashwini
trust
said,
it.
after collection, what is the duration? Before, I mean, we say that we have kept it for liquefaction, but then whether you have kept it at room temperature first, then at 37 degrees centigrade, how long at 37 degrees centigrade, and most importantly, have you centrifuged at a high speed at 37 degrees centigrade? I mean, the sperm capacitation.
I mean,
itself
there are two school
will
of
be
thoughts.
affected.
There are two school of thoughts. People say that we have to allow them to liquefy at room temperature, and some say at 37. Similarly, with even centrifugation, there's only one centrifuge, one or two in the country, I would like to say, where they used 37 degrees Celsius for the centrifugation process. Otherwise, it is basically the RPM, which is a room temperature based RPM centrifuge, what we use. So I've done
Many companies have sold centrifuges, sperm fuges, which saying that at 37 degrees centigrade you have to centrifuge.
Yeah.
No, it's not needed. Technically,
It's not needed.
Not needed.
Because the very RPM itself is going to create the
I was in the
temperature.
process of
Even RPM depend upon the centrifuge model and the
yes,
whatever it
Angle of rotation.
So that
Angle
is
of
also
rotation.
it is not you have to see that swing down model, swing down model. There are so
No,
many
no,
variations in
you
the
have a higher
absolutely
at a
yeah
lower RPM.
absolutely.
Swing out basically we use for DNA
Rotors will
extraction.
have lesser RPM at a higher rotation.
So many things. It is not only that we are the 37 we are checking, but yeah.
I think as we
Triangle
are
of
careful with
rotation,
the
triangle
oocyte,
of
we should be
preparation,
careful
so I
with
know
the
that.
sperm also.
Well, having said that, I mean, to the last question for the day. So in case of male factor infertility with abnormal sperm morphology, so how does this underlying cytoskeletal defects?
XA for all.
No, ma'am. I mean to say with respect to even the stunt tails of spermatozoa, where there is if you look at it, there are genetic defects in that. So which are directly correlated to the ciliary moments of the trachea. I mean, that gonna reflect in the sperm as well. So as such cases, which are genetically indicated, how can we mitigate this? We know that it has a direct effect on the outcome.
See, you cannot mitigate this, first of all.
Yeah.
It is there, it is there. So don't select it if you are doing it. See, if you are doing IVF.
What are the tests do you recommend, sir? Like, genetic tests, what are the tests that you're going to recommend for the parents as well?
There is no test here. Even if you do the test, can you repair it? It is not possible.
It cannot be done. So the ideal recommendation would be?
Donor
In case if you are doing ICSI, then do not select them. Spend your time to select the sperms
the
first,
sperm, yeah,
make them into a place, and then take the oocyte and take the injection. That is first. In case of an IVF, depend only on double density gradient technique. Even you can make triple density gradient. Don't depend on any other methods.
So you mean to say you harvest the sperm first, take your own time, sweet time to harvest the sperm, and then once the harvest is done, you can get the oocytes and inject.
You can get the oocytes out and inject with the harvested sperms, and always select more than required sperms. Say if you have got 10 oocytes, take at least 15-20 sperms.
But that is the common practice for all TISA, micro
No,
TISA,
not always.
No, I do. If the
Not
sperms
always.
because
I have seen some people who have taken, the TISA macerate directly onto a dish, and they are taking it from there, calling the junior, okay, give me the oocyte dish. I am injecting this. So let's not go into that particular aspect of it.
I mean, all said and that, sir, for the juniors, I think it is not recommended. You get them into PVP wash drop, immobilize them, give a neat wash, because there are a lot of other factors that gonna have impact on the embryo formation. So you have to wash them off, get a clean sperm before injection.
Yes.
What recommended. And yeah, having said that, I mean, that's the end of the panel discussion. I mean,
there is one thing I have like to know. I don't know whether somebody, Bindu or Vijay, has done or not. In earlier days, we used to take cumulus into capillary and keep it into the process sperm at 45 degrees centigrade, and the sperm used to migrate. Then used to cut the capillary from and take the sperm for the ICSI.
Outdated technique, ma'am. We were not.
No, that is not needed. That is not needed. There is a definitely
glass wool
method
wala.
of testing.
The glass wool technique wala.
Not glass wool. Not glass wool.
Okay.
ये इसको करते हैं ना, clotting time.
This is zona binding. Zona binding
Correct.
Capillary wala,
that was our own way of finding out whether there is zona binding.
Yeah.
I mean, nobody has measured the temperature of ampullary region of fallopian tube, nor the.
See, the core temperature is usually 0.5 degrees higher.
I mean, right. Sir.
Number one. Number two, where the action is occurring, you have to understand that the fallopian tube has got ciliary epithelia internally. So if the ampulla has captured the oocyte, and if it is transmitting it towards this one, naturally work is occurring more, so it will be having a little higher temperature gradient.
Correct.
And the way sperm moves to that is by heat-seeking mechanism only.
So slightly higher means say 0.1 or 0.2, not more than that.
That's what I'm saying. Slightly higher gradient than the rest of the uterus.
Sir,
that particular part where it is moved.
No, chemotaxis, rheotaxis, geotaxis, and
Geotaxis,
also the
it is thermotaxis. It is thermotaxis.
Right. Having said that, I mean, that's the end of the discussion. I think we can go on like this, because this has brought multiple Nobel Prizes on the very topic cytoskeleton. I mean, this never-ending story. It keeps giving Nobel Prizes as the year goes on. I mean, yeah, thank you so much for all the panelists. Had a lovely time.
Thank you.
Thank you.
Thank you so much, Sanket. We too had a lovely time.
Yes, yes.
Bye.
Lots of insights.
एक picture ले ले.
Yeah. Charu sir is, I mean, your camera has to be on.
Charu is here.
Sir.
Charu, where are you? Charu. Yes. थोड़ा light ले लो. थोड़ा light ले लो.
नहीं. Mera Sanket, you have missed a very important point in today's this thing.
Hello, sir.
You have not talked about anything about pH, temperature of embryologists while working.
Yeah. That is the most detrimental thing, because we will be having
the embryologist.
The physical, mental status of the embryologist, pH, temperature of embryologist.
It will show a very high reading of ROS, sir.
Hunger status of embryologist.
Sir, media चाहिए.
Nakul, can you stop that?
मेरा एक बहुत अच्छा लेक्चर तैयार है इस पर. Stress on embryology.
Live streaming stop कर दो, please.
Are we offline? Nakul, are we offline?
Yeah, please stop the recording.
Yeah, please stop. Take us off.
Recording and as well as live streaming.
मैडम, यदि आपका लेक्चर स्ट्रेस
Yeah,
ऑफ
Ankit, please stop the recording.
stopped.
तैयार है
Yeah, sir. 1 minute बीच में हाँ.
Yeah, we are
मैं
offline.
कहूँगा कि
Cool.
यदि
Yes.
आप